Okay, good morning, everyone. I think all the clicks and buttons are on, more or less. So we will go with our second lecture on the bioethical issues at the beginning of human life, beginning always with a little prayer in the name of the Father, the Son, and the Holy Spirit. Blessed Trinity, we love you, we adore you, and we thank you for your many blessings, for the gift of life, and for having us here today once again by your divine grace. We ask that you inspire us, Lord, to go deeply into our human nature, and especially this issue of when we begin individually, so that we may uphold the dignity and the sanctity of human life precisely at every stage of development, and then go forward and share that knowledge with those who surround us. We pray in a very special way today for those who are in most need of your divine mercy. In the name of Christ our Lord. Amen. Father, Son, Holy Spirit.
Do you have any handouts for us, Father?
Yes, I left them on the photocopy, so give me a moment. I'm going to pause this as I go to the photocopy machine where I forgot the handouts. Excuse me. Okay, so the handout is the outline, the same outline that I sent by email yesterday. Essentially, and I added a little section at the bottom here, so I'll resend it. But it's basically some lines toward the end on the actual, what we call the ontological status of the human embryo. This third point here, and I'll cover that toward the end of the lecture today. Okay, and I'll send the updated outline together with the video at the end of the lecture. Okay, so any questions or comments from previous? No? People are good. We covered a lot of material, mostly the biology of DNA and asexual reproduction, and the whole, what is known as the central dogma in biology, from DNA to RNA, and from RNA to proteins. Proteins are the ones that do the work in the cell, what we call metabolism. And so there we have the whole package. First of all, with DNA, we have the molecule of inheritance passed on from one generation to the next of whatever species. It is the universal genetic code, if you will, the universal molecule of inheritance, DNA. And then DNA stays in the nucleus, protected there, and sends out the message so that proteins will do the work, what we call metabolism, of the whole organism. And that applies universally, meaning from single-cell organisms like amoeba and paramecium to huge, large, macroscopic plants and animals like you and I, and whales, and all the living species that exist. Okay, so it's a very elegant system. When you think about it, it's really very simple to keep life going at the organic level on this earth, generation after generation. And there is that general propensity in nature to duplicate, to replicate, to reproduce, and to maintain the species going, and not just going, but thriving, actually, thriving. Because as you recall from last semester, one of those points that Meyer made with regards to evolution, which was actually one of the observations of Darwin, and that made him think about evolution and adaptation, is the overabundance of the production of progeny, right? Now we're seeing at the biochemical level how that progeny comes around and maintains the same species, maintains the same species. It's all because of the genetic code and then the deciphering of that code into functional proteins. Okay, so that's kind of a summary, big picture. As I was going over the homework and going over the summary from last lecture, I think I forgot to mention the actual level of compaction on the supercoiling, right? From that 2-meter strand of DNA, if we stretch it out of every single cell, to the super macroscopic chromatin or even more condensed chromosomes, the level of compaction actually is, anybody have an idea? the level of compaction, the folding of the folding, that coiling that I spoke about, is about 10,000 fold, 10,000 folds. This is what I mean. A normal sheet of paper, 8.5 by 11 inches. Anyone know how many times it can be folded normally without any machinery? Huh? About 8 times, right? So here is half. So that's 1 fold, 2 folds, goes into quarters, right? 3 folds, goes into eighths. 4 fold, 5 fold, actually 6. I cannot fold that any further. If anyone can do that with their fingers, you're welcome to try. That's 6 fold, all right? That's like we would say this would be equivalent to a 6 fold compaction of a normal piece of paper. 6 fold. Now 10,000 fold. That's how one can fit 2 meters into the nucleus of a macroscopic cell. All right, and that's what nature does. That's what we call it, simple and elegant. All right, that was the outline of today, by the way. Anyway, let's go forward. Today, we're going to look at the other mode of reproduction, sexual reproduction, and also early embryonic development in some level of detail, and hopefully we'll end with the bioethical analysis, all right, which is what is the ontological status of the human embryo? In other words, what or who is the human embryo, and when does the human embryo actually become human? All right, you can see that's a philosophical, theological question, and that is what we mean by the ontological status, the being, ontos meaning the being itself. What is this being that we're calling the human embryo? Okay. Moving forward then, so first we're going to get into sexual reproduction. Now, sexual reproduction involves fertilization, in contrast to asexual reproduction, where it's just the one individual reproducing by whatever means we talked about last time, fusion or segmentation or parthenogenesis, etc. In contrast to that, sexual reproduction involves fertilization. In other words, involves the fusion of gametes. Fusion, the coming together of sex cells that we call gametes. So what happens with fertilization is that the diploid number is restored on the new individual. Okay, the diploid number is restored on the new individual, but this time it's done by the fusion of 2 cells, of the sex cells that we call gametes, which are not diploid, they are haploid. In other words, they don't have a double set of chromosomes, just a single set. So the gametes are the sex cells, which are also known as sperm and ova, sperm for male, ova for females. 1 ovum, several ova, that's the Latin. And the way that these gametes are produced, are generated in nature, is a process called meiosis. And meiosis is a particular type, it's like a double cell split, it's like a double mitosis. But in that double mitosis, something very special happens to the number of chromosomes, okay? And to the quality of the chromosomes themselves. So we'll get into that. But for now, this issue of meiosis is a double splitting of the cell, so that from 1 we go to 2, and from 2 we go to 4. Okay, 4 daughter cells from a single mother cell, in contrast to normal mitosis, which is just 2 daughter cells from a single mother cell. All right, and because meiosis is somewhat of a double mitosis, all right, I just want to recap mitosis for a moment to some level of detail and use a diagram, pick up where I left off last time, which is this diagram. I want to make this diagram a little better, make it more clear. Remember, we start with the cell at interphase, where the cell is metabolizing, doing its normal function, right? And there are no distinct chromosomes yet. It's called chromatin, a single strand just supercoiled into its functional unit. And What I'm going to do is I'm going to relabel these chromosomes of the prophase mitosis. So instead of just A, B, C, D, I'm going to give them a little more informative number, okay, which is this. I'm going to label them actually how they would normally be labeled in nature, in science, which is by size, all right, by size. So how many total number of chromosomes do we have here?
Yeah, 4 pairs.
4, okay, or 2 pairs, 2 pairs, all right? And let's keep it simple for now, 2 pairs. There is a large pair, and then there is a small pair, okay? So the large pair, I'm labeling as chromosome number 1 because it's larger than 2. And that's actually how they are labeled, these 23 chromosomes on the human, chromosome pairs, when one does a karyotyping, what is known as karyotyping. All right, don't worry about the word for now, but let me show you karyotyping of the human chromosomes. It's just spreading out the chromosomes by image, okay, and they're numbered. Let's put this one, it's going to be small. Let's try to make it larger. Yeah, okay, so here is 22 plus the XY, that's the 23 pairs of chromosome on the human. And we know right away from looking at this a couple of things. We know that this is indeed human because it's 23 pairs. Also the size, I'm just pointing out, you see that number 1 is much larger than number 22, right? And so they're just numbered by size basically, and then paired up. Again, by size, so you would not pair up this one with this one, all right? You would pair them by size. And then we also know it's informative, this karyotyping is giving us a 23rd pair, XY. So we can tell the sex of this individual is male, all right? The sex is male because it has a Y chromosome as opposed to 2 X chromosomes. All right, so just to point out that the chromosomes are numbered according to their size in whatever species when a karyotyping is done. For some reason, they have labeled 18 here. I don't know why, but it's not relevant to our lecture. All right. So you see I'm labeling chromosome 1 as the larger one and 2 smaller. Got it? Then just to distinguish one pair from the other, A and B. A and B. And the coloring actually is also indicative, I will say, in anticipation that the coloring means that one of the pair comes from the father and the other of the pair actually comes from the mother, okay, from the gametes. So 1A came from the sperm of the father and 1B came from the egg of the mother. And the same with 2A and 2B. That's why they're just painted green and pink.
So they paired by size.
So by size, the number is by size, okay? And the pairing is according to whether it comes from the sperm or the egg, all right? Anyway, this is a little more informative, this type of labeling, and so that's how I'm labeling them. Also, I want to complete this diagram, actually, go beyond telophase, back into interphase. So here it is. A little smaller because I have to fit a new interphase here, just to show that from the original mother cell in interphase through mitosis, we end up with 2 daughter cells, okay? Very straightforward, you're all with me, right? Okay, let's go a little more level of detail and actually let's see what happens here from interphase to prophase, just a very brief review. Here are the 4 chromosomes that I had labeled earlier. Here is, actually, I need to relabel these. I forgot to change them. What's wrong with this diagram? I forgot to put 1 as the biggest one and 2 as the smallest one. Originally, I had them backwards. Come on. Sorry. Oh, I think I got it now. Please check, make sure that I got it. The big one is 1, and the small one is 2. So 1A corresponds to this one here, and 1B, right? This is the cell at interface, but getting ready to duplicate, getting ready to undergo mitosis. You notice then that is indicating, of course, the cell is diploid because chromosomes are in pairs, right? And it's indicating here in this little diagram going before and after, before and after. What is this before and after? What is happening? In interface, but preparing for replication, preparing for the cell splitting and duplicating. Yes, John? Mm-hmm.
The cell replicates its DNA and made the sister chromatids.
Exactly. So this is the replication here. So this arrow is indicating a replication, which we know already. And just like John said, these are the sister chromatids, which are attached in the middle. So I can put a little more labels here. All right. The arrow indicates DNA replication or duplication. Okay. And so each one of these legs is known as the sister chromatids, and the chromatids are attached more or less in the middle somewhere by a region called the centromere. Don't have to worry about that level of detail, but that's the duplication there. Okay, each sister chromatid is a duplicate of the original single strand, single DNA double helix. All right, so you're with me, okay? Mm-hmm. All right. This is just kind of a review of last time. And so I can show you now where is that temporary 4N stage, all right? The temporary 4N stage is in prophase and metaphase, in that we can say that we have 2 pairs of chromosomes here, right? Pair 1 and pair 2, total of 4 chromosomes. But a little more technical, really, how many chromatids do we have, sister chromatids, in this prophase? We have 8 sister chromatids, right? And so that makes it the 4N, because what is the N number? How much is N? Is the number of chromosomes. And how many chromosomes originally? Chromosome 1 and chromosome 2, right? There are 2 chromosomes. So N, in this case, is 2. 2 times 4 is? 2 times 4 is? 8. And that's the 8 chromatids, sister chromatids that we have here in total. Okay? So that's that intermediate 4N stage that we talked about. Doing prophase and still in metaphase because the sister chromatids have not separated yet. Then when we go from metaphase to anaphase, you see how the sister chromatids separate. In other words, the centromere gives way. The centromere gives way, and each individual sister chromatid is pulled away to the opposite pole by these fibers, which I told you was another type of mostly protein machinery, okay? And they're being pulled to the opposite poles, to this little area called the aster. Too much detail, but notice how the sister chromatids line up here in pairs, and then they're split. That centromere, that middle region, gives way to the pulling in opposite direction. So be clear about the numbers, the structures, and the mechanism from stage to stage. Questions? No? People are good? So begin with N is the number of chromosomes. Well, we got chromosome 1, we got chromosome 2. They're paired because it's diploid. So 1 has a pair, 1A and 1B. 2 has a pair, 2A and 2B. And here, when you see sister chromatids like that, in other words, when you see the chromosome looking like a little X, that means that replication has already occurred. And that cell is ready for mitosis, for splitting. Okay, forward. Now we can talk about the cell cycle as a whole. The cell cycle as a whole, okay? And you know, remember the cell has to cycle. Why? Because as the cell metabolizes, the cell grows. As the cell grows, it runs into that volume issue, that volume problem, that the membrane is getting too taut, is getting too stretched out, so it has to split to favor the surface ratio.
So where are we at now, Father?
Okay.
Are we moving into telophase?
Right. So in the cell cycle, you can see that it's mostly interphase. Okay, the cell cycle is mostly interphase, what is indicated in blue here. But when the cell goes into splitting mode, it goes into the mitosis or mitotic mode, is this section here. And then you have the two resulting daughter cells. Each daughter cell will then continue to metabolize, start growing again, and go through the same cycle. What is not indicated is the size, that these two daughter cells will originally be smaller, and then they'll grow larger into that volume issue. Okay, so let's stick with this cycle for now. Like I say, blue is the interphase, and just for technical reasons, it's subdivided into 3 stages. What is called G1, the G comes from gap, which some people also interpret as growth 1 and growth 2, but originally that G comes from just the first gap of the cell, first gap in the cell growth, cell metabolizing, all right? During G1, what happens is that all the little organelles inside the cell are duplicated except for the nucleus. And then the cell goes into this S phase. S stands for synthesis. And what is being synthesized? What is being synthesized is the DNA, okay? So the word is a little awkward because you would think that synthesis is crunching together, but actually in this, this word synthesis is used in the chemical meaning, in the biochemical meaning of replication or duplication. So synthesis in chemistry means making another product, making a new product. That's what synthesis means, okay? In chemistry, is making a new product. It's a chemical reaction. So this S phase is, we know it as replication, when the chromosomes duplicate, all right? And you can see it here, for example. Here are the 2 little chromosomes, okay? One blue, one red, and now they have duplicated. So now you can see the sister chromatids in that typical X fashion, X representation. Then the cell goes into a second gap phase known as G2, where it's like a double check. Here the cell makes sure that the DNA has already duplicated and that also the organelles in the cytoplasm have also duplicated so that the cell is ready to go now into mitosis. All right. So there is this is the interphase part, and then mitosis itself with its 4 different stages: prophase, metaphase, anaphase, and telophase, which is all gradual, okay? And the end result is the 2 daughter cells. Now, periodically, a daughter cell, as it metabolizes and grows and specializes, right, becomes specialized into particular tissues of the body, periodically one of these cells goes into a mode of arrest. In other words, it doesn't duplicate anymore, doesn't duplicate. The cell cycle arrests, and that's called G0 or GO. And some of these cells are typically super specialized cells in the body that do not replicate anymore, generally like neurons, for example. It's a classical case, okay? And some other cells that become super specialized and they just hang out, they don't replicate anymore. Okay, so that's the cell cycle. The interphase is subdivided into G1, S, and G2. And the most important aspect for our concern is this S phase, where the chromosomes are duplicated. Questions, comments? People are good. We go forward. All right, now let's look at meiosis. So again, that was a review of the standard mitosis. I'm telling you that meiosis is like a double mitosis with a twist, and the twist is going to be on the first doubling, all right, early on this first doubling. But here we have that meiosis is a double split. So from one original cell, we're going to get eventually 4 daughter cells. 2 and then 2 again. And during this process of meiosis, 2 things happen that are very unique and very important and essential precisely to form the gametes. One is called recombination or crossing over, and this recombination or crossing over would happen at the level of individual chromosomes, actually at the level of sister chromatids, the sister chromatids. I'll explain a little further what that is about. The other thing that happens is that the number of chromosomes is halved, all right? So that from the original diploid cell, we're going to end up with a haploid number. Diploid meaning 2n, the prefix di is what indicates 2. The I is diploid is 2n. Haploid, HA means single, okay? HA, the prefix HA. So a single number, in other words, 1n, and the coefficient 1 is usually not shown, so just n number. Means that there are no pairs. What does this mean functionally? That the chromosomes are singled out, they're not paired. Okay, this process is called chromatic reduction. Chromatic reduction. Right? Chromatic, a reference to the chromosomes, and reduction meaning that there will be no pairs, that the set will be single set. Forward. Here is my meiosis summarized, beginning with interphase, a diagram that we're familiar with, that I used. Here is the cell in interphase. You can see the individual chromosomes. Again, how many chromosomes here? How many chromosomes? Total number of chromosomes? 4 chromosomes. 1, 2, 3, 4. How many chromosome pairs? 2. All right. So this cell is diploid because there are pairs of chromosomes. How much is n? The value of n. The value of n is 2, right? Chromosome 1 and chromosome 2 are paired. All right, so that makes it diploid. All right. Okay, and this is showing replication. So we already have sister chromatids in the typical X shape. Now, the cell goes into this double split, which is known as meiosis 1 and meiosis 2. Meiosis 1 and meiosis 2. All right, so in meiosis 1, this is prophase 1, metaphase 1, anaphase 1, telophase 1. And then prophase 2, metaphase 2, anaphase 2, telophase 2. It's all very systematic, very methodic, and every meiosis undergoes all of these steps. So it's a total of 4 plus 4, a total of 8 steps, from 1 cell to 4 cells at the end. You notice that the 4 cells at the very end are haploid. So where does the halving, where does the split in half happen? Where are the chromosomes singled out? The key is here, between prophase 1 and metaphase 1. Okay, notice this, and we'll go into a little more detail in a minute, but notice. So this represents 1 chromosome that has already replicated. In other words, 2 sister chromatids, right? Attached by the centromere that I mentioned. Look at here. You notice how these sister chromatids have stayed together with their pair. So I mentioned earlier that blue was indicating the father's DNA and red or pink was indicating the mother's DNA. All right, you notice how they stay paired up. So these two sister chromatids, let's say this is chromosome 1, is paired with the sister chromatids, both sister chromatids of chromosome 1 of the pair. Okay, so you have double pairs here. The first pair is the two sister chromatids, which are held together by that centromere. And the second pair is chromosome number 1 pair. Chromosome number 1 pair, and therefore chromosome number 2 also pair. Okay?
We call this homologous, right?
Yes, exactly, thank you. These chromosome pairs are called homologous because they have the same size. They have, in general, they will have the same genes, all right? They will have the same general genes, which we call alleles. All right, so these are homologous chromosomes. For example, chromosome 1A, let me clarify further, and thank you for the information. So chromosome 1A and chromosome 1B are homologous. And chromosome 2A and chromosome 2B are also homologous. Let me write it in here for a moment since I'm mentioning the word. Okay. Let's just stretch this out somehow.
Exactly. And you said, Father, called chromology just because they appeared by size?
Yes, exactly. These are chromosome pairs. Same size. Okay, stretch it out. Okay, all right. So 1A and 1B are homologous, 2A and 2B are homologous, but 1A and 1B, 1A and 2A, for example, they're not homologous. 1A and 2A, right, are not homologous. 1A and 2B are not homologous. 2B and 1A and 1B are not homologous. In other words, chromosome 1 and chromosome 2 have different sizes. And therefore, they're not homologous. If they're not homologous, then heterologous. Don't worry about the name, it's getting too much, okay? But if they're not homo, they're hetero. Okay. Back to meiosis. Here we go. Okay, so the homologous pairs stick together. That's a good way of putting it. Homologous pairs stick together with all the sister chromatids still attached. So here we have homologous pair 1 and homologous pair 2. In the end, how many total number of chromatids do we have in each cluster? Total number of chromatids? 4, exactly, 4, all right? 2 from one of the pair and 2 from the other, that's 4. So that's called the tetrad. Those are the famous tetrads. Those are the tetrads. See, and so the tetrads stick together in meiosis 1 at the very beginning, at the prophase of meiosis 1. Those tetrads stick together from prophase to metaphase, so that when the chromosomes line up at the equator of the cell, and you notice this cell now is turned horizontally, it doesn't matter which way it's rotated, the equator is always going to be in the center, or also known as the mitotic plate, or the middle, the metaphase plate, right? When the chromosome pairs, when the tetrads line up, they line up as tetrads precisely, as those double pairs. So that when these double pairs start migrating, they separate and they migrate to the poles in the third stage, in anaphase. They migrate as pairs. They, the sister chromatids. Sister chromatids migrate as pairs. In other words, the centromere does not give way, all right? And that's the crucial moment. That's how the number of chromosomes is turned into half. That's how the diploidy is lost. In that, in meiosis, the sister chromatids migrate to the opposite poles as pairs. The centromere does not give way. That's the crucial step here. From M1 to A1, that is the essential crucial step. contrast this step to up here in mitosis, where you see the sister chromatids line up, right? But do you see any tetrads here? There are no tetrads. You see, here's 1A, here's 1B. They're independent. There are no tetrads. And 2A and 2B, so that when the sister chromatids migrate to the opposite pole in mitosis, the centromere gives way, and the sister chromatids migrate individually. So that's the key here, the difference between mitosis and meiosis, in that in meiosis, the sister chromatids migrate to the opposite poles as pairs, okay? And so to complete that first cycle, this cell, the mother cell, will undergo cytokinesis, in other words, splitting in the middle. The nuclear membrane is reformed on the sister chromatid pairs. But then it continues into meiosis 2. Each one of these two, now we have two daughter cells, right? One cell division has occurred. We have these two daughter cells. Each one of these two daughter cells will now essentially undergo mitosis, normal mitosis, okay? So here you have one daughter cell. The sister chromatids line up at the equator as pairs, and then they split. The pair splits when they're pulled to the opposite poles. And finally, the two new daughter cells are generated. And it happens twice because there were two original daughter cells, ending up with a number of four daughter cells. So really, meiosis 2 is a mitosis. Meiosis 1 is early on. Here is the trick here. Here is the detail, okay? This tetrad formation. Let's look at a diagram now where it has parallel in stages, normal mitosis, and then the special case of meiosis. Let's start with our cell, original cell in interphase. All right, this cell is diploid, meaning 2N. How many number of chromosomes do we have in this diploid cell? We have 4 chromosomes, right? 1A, 1B, 2A, 2B. There are 2 pairs. Chromosomes are paired because it's a diploid number. So it's a total of 4 number of chromosomes. All right. Now, if the cell undergoes mitosis here on the left, what happens from here to here? Replication, right? Because you notice that the chromosomes are looking in that typical X fashion, the X shape. So the X shape means that they're sister chromatids. To get sister chromatids, these original chromosomes had to duplicate or replicate. So what happens from here to here is replication in that S phase of the cell, the synthesis phase. And now this cell is ready to undergo mitosis. So the sister chromatids line up at the equator. Here's the equator. It's put horizontal, okay? Here's the equator, and here are the sister chromatids in pairs, but no tetrads, no tetrads. So 1A is independent of 1B, and 2A is independent of 2B. And actually, if I had improved, this diagram can be improved 1 notch. This diagram could have been improved by just switching the order of these 2 chromosomes, for example, so that it is abundantly clear that 1A and 1B are lining up independently of each other. To make this diagram just a little better, I would have put like 1A over here and 2B down here and mix this, so not put it in sequence like that. You see what I'm saying? Because they're lining up, the sister chromatids are lining up independent of the chromosome pair. And then when they are pulled, when the sister chromatids are pulled to the opposite poles, the centromere gives way, and you end up with individual sister chromatids in the daughter cells, which become the new chromosomes because they are a copy. Remember, they are a duplicate. They are a copy of the original chromosome. Simple mitosis. In contrast, meiosis 1, you see, this is all meiosis 1, meiosis 2 is all down here. Meiosis 1, the sister chromatids line up. First, they make the tetrad. They make the tetrad, so the pairs of chromosomes, 1B and 1A stick together, 2A and 2B stick together. They form that tetrad. They line up as tetrads, and when they do the first split, what splits is not the centromere, but what splits is they stop recombination, and they migrate as pairs, Then these two daughter cells, like I say, they undergo normal mitosis. You see that this diagram is similar to this one, only that this one ended up with the maternal genes and this one ended up with the paternal genes. They undergo mitosis and you end up with the 4, but now these cells are haploid, because the split happened through meiosis 1, so that one set of genes went to one cell and the other set to the other cell. Okay, and so bottom line, in meiosis 1, this is where the chromatic reduction occurs from diploid to haploid. Okay, question, comments? No? All right. Review the diagrams, but the key, again, is keeping in mind this tetrad issue. Tetrad, which forms the chromosome pairs, homologous chromosomes by size. Now, I mentioned, so what this does is that chromatic reduction going from 2N to 1N, right? From diploid to haploid. Another event that happens, which is kind of associated down to the DNA level, is what we call recombination or crossing over. This also adds variety to the next generation. So it's one of the advantages of sexual reproduction. You may recall earlier I mentioned that why undergo sexual reproduction energetically is twice the effort. It's twice the effort energetically on average for an organism to produce gametes and to undergo fertilization as opposed to asexual reproduction. So there has to be some advantage to counterbalance that extra energy expenditure, all right, in nature. And the counterbalance is that it allows for variety. Sexual reproduction allows for variation to occur in the next generation. In contrast to asexual reproduction, where in asexual reproduction, the next generation is identical. They're essentially clones because one is starting with a single cell or a single individual, and from that one individual's DNA comes the next individual. Whereas in sexual reproduction, the new individual comes from the fertilization, from the union of two previous individuals. So it allows for variation. Actually, variation at two levels. At the level of this recombination and crossing over, and also at the level of the fertilization itself. You'll see what I mean in a minute. So let's look at recombination then. You're familiar with this diagram, these X's. And what does the X, as soon as you see an X like this, chromosome represented as an X, what does it mean? Sister chromatids, right? Sister chromatids attached by the centromere, meaning that they have duplicated already, that the chromosomes have replicated. And this side of the chromosome should be identical, almost identical, 99.999% identical to this one, okay? And then these are pairs. You see they're the same size, right? So these are homologous pairs. You got two sister chromosomes. This could be 1A, this could be 1B. The colors are just representing, again, if they come from the father or the mother, meaning from the sperm or the egg. This is recombination here. This is recombination. Remember that they are together. They're close together. In close proximity, they actually line up like that, the two pairs, in that tetrad formation, They have that tetrad formation. And in that tetrad formation, some of the legs cross over and recombine. This crossing over is a complex biochemical process that is all guided by protein machinery, very specific, very precise. The machinery knows what it has to do according to the actual sequence on the coil here, on the supercoil, all right? But functionally what happens is that these legs cross over so that a piece of the blue leg will end up on the red chromosome and a piece of the red leg will end up on the blue chromosome. And this is the end result of crossing over. All right? So the legs that actually crossed over are called recombinant chromatids because they recombined. The legs that did not cross over, as you can see here, they're called non-recombinant. Too much level of detail for the wording, rather just interested in you understanding that when the sister chromatids line up as tetrads, that's when crossing over occurs. And crossing over means that they are exchanging homologous pieces of information. Homologous pieces of information that are, like I say, 99.999% identical, but it's in that 0.0001 difference that makes for variety. Let me give you an example. Let's say that hypothetically, the blue chromosome comes from the father, meaning the sperm that fertilized that particular egg, and the red chromosome represents the mother, meaning it was in the egg, in the ovum, okay? And let's say that this is chromosome 1. The largest one. So this would be 1A, this would be 1B, you're with me, all right? And let's say that in this region, the bottom region of these legs is the code for color of hair, okay? And let's say that the father had brown hair. So you would have the brown genes down here, brown hair. And let's say that the mother had blonde hair, okay? Does that make a different species? No, it's just variety, right? Remember quantitative traits? Color of hair is a variety. Or we can make it size. I think it's simpler with size. Let me do it with size, actually. Let's say that the father was 6 feet and the mother was 5 feet. Exaggerating on both ends. All right. All right. And the height genes, simplifying, are at the bottom of the legs here. Okay. So chromosome 1 is the height gene, the gene for tallness, and the gene for not so tallness, which we will call shortness. Father 6 feet, mother 5 feet. When they recombine like this, all right, what do you expect the child to have? The child would have 5 feet. I'm just saying on average, the child would have 5 feet if the child receives either this gene or this gene. But if the child receives this gene, the child would have 6 feet. And if the child receives this gene, the child would have 5 feet. Okay, now of course, if the child receives this gene, it would have to be a boy. And if the child received this gene, it would have to be a girl, because this is a female chromosome and this is a male chromosome. But here, either boy or girl could be, actually this boy could be 5 feet and this girl could be 5.5 and 5.5. So simplifying the issue with crossing over, what happens is that the variety of one gene goes into the other chromosome. And the variety of the other gene goes into the one chromosome. Okay? So it's exchanging varieties of alleles. It's subtle but real, because remember, most traits are quantitative traits. In other words, they're on a range from the biggest to the smallest, the strongest to the weakest, the tallest to the shortest, etc., etc. Quantitative traits, right? And that's what happens biochemically with recombination at the level of crossover, because eventually, you know, this tetrad will then split, and each one of these 4 chromatids is going to end up in a different daughter cell. So the 4 daughter cells that will result from this particular chromosome. One daughter cell we have fully blue, the other daughter cell we have mostly blue, some red. The next daughter cell we have mostly red and some blue, and the fourth daughter cell we have all red. So the 4 daughter cells are slightly different from each other. Those daughter cells, they're gametes, so they're either sperm or eggs. And you will now have the variety in the sperm or the egg. Slight variety, but real variety. Okay? So crossing over introduces variation on the next generation. Now, let's line them up. The diagram, which we're not familiar with, line it up with the other diagram from meiosis 1, right? And here is the tetrad. What is represented big here between the blue and the red chromosomes, that's a tetrad, because this is 1A, this is 1B, right? So here's the tetrad, and this is another tetrad, smaller chromosome. So you see, here's the recombination shown. I just magnified a little bit so you see the recombination that is being shown here. Okay, see the recombination there? And then when they finally split, the recombinants, one of the sister chromatids is recombinant and the other one not. Okay, and the same on the other pair. And here is showing 2 sets of chromosomes instead of just 1 set of chromosomes in this diagram. You follow? So that's the 2 events of meiosis. There's chromatic reduction from diploid to haploid, and there is recombination or variety, mix. Forward. Okay. Anything on the diagrams? People are good? All right? All right, let's go forward. So meiosis is formation of gametes, which are the sex cells, and you know already, haploid. Now, it turns out that meiosis is a general term, but really in nature, functionally, it's called gametogenesis. In other words, the formation of gametes. Essentially, what I'm doing is explaining the vocabulary to you so that if you're not, if you don't have that scientific background, then you will be competent to address the issues with the proper language. Okay, so gametogenesis is the formation of egg and sperm. It's just a fancy word for making eggs in the ovaries or making sperm in the testicles. And therefore, gametogenesis is either spermatogenesis, if it's making sperm, or oogenesis, if it's making eggs. And N is just symbolizing the haploid number. Forward. Again, back to a diagram. But now in the context of gametogenesis, all right? So you'll see some terms. There's too much information here, so don't worry about it too much. I just want to show you really the end result, which is very important because the end result will be different for sperm or egg as far as the number is concerned. You'll see what I mean. Now we start with our diploid cell, okay? These 2 diagrams should be familiar to you, these 2 figures. What do you see? You see excess, that means sister chromatids attached by the centromere, so replication has already occurred. How many pairs of chromosomes here in these original cells? How many pairs? 2 pairs. Total number of chromosomes 4. Total number of chromatids 8. All right, 2, 4, 8. All right. Now, in the case of spermatogenesis, the formation of sperm, this diploid cell inside the testicles is called the primary spermatocyte. The primary spermatocyte site is a reference to cell, spermato because it's going to be a sperm, and primary because it's diploid. All right, is the cell that generates the sperm. In contrast, in the egg, in the ovary, it will be a primary oocyte, again, diploid. Then the cell goes into meiosis. So the tetrads line up at the equator. Here's tetrad 1, here's tetrad 2, tetrad 1, tetrad 2. You see the tetrads lined up, okay? Meiosis 1, first meiotic division. The sister chromatids migrate attached in pairs. So here's sister 1, here's sister 1, chromosome 2 with the sister chromatin pairs. Meiosis 2 is that simple division where the centromere splits, so one chromatid is in one, the other one in the other, and the same thing down here. You end up with 4 daughter cells at the end of meiosis 2, all right, the 4 daughter cells. Let me stop there for a moment. What is not shown from here to here, from meiosis 1 to meiosis 2, what is not shown in this meiosis 1 that I just talked about?
Crossing over.
Exactly. What is not shown is crossing over or recombination. It will occur here, right? When the tetrad is still intact and still in place. It's not showing crossing over. For the sake of simplicity, they're trying to keep the diagram simple. The end result is 4 daughter cells in spermatogenesis, which eventually mature into sperm cells. They grow a tail, spermatozoa. They mature into sperm cells. In contrast, what happens in oogenesis is that the split in meiosis 1 is not an even split. In other words, one of the 2 daughter cells maintains most of the cytoplasm. Most of the cytoplasm. You can think of it as selfish, I don't care. But the other daughter cell is deprived of cytoplasm, too little cytoplasm, okay? Those 2 daughter cells undergo mitosis the second time around, and that one daughter cell does the same thing again. That one daughter cell hugs most of the cytoplasm again. So that you end up with these 3 little tiny minute cells that don't really have enough cytoplasm to be functional. And therefore they degenerate and actually become attached. Sometimes they get reabsorbed. These get reabsorbed. This one gets attached here. And they're called polar bodies. Polar bodies because they don't really have enough cytoplasm to be functional. And therefore, functionally, the end result of oogenesis is that you end up with 1 daughter cell, which we call the ovum or the egg, and is haploid because it hugged most of the cytoplasm out of the other 3 daughter cells that become degenerate. They're called polar bodies and are either reabsorbed or they just stay stuck. This one actually stays stuck here like a little dot, all crunched up against the inner membrane of the cell, okay? And so in general, spermatogenesis and oogenesis begin with diploid cells in the testicles or ovaries, but at the end, we end up with many more sperms, 4 times more sperm on average than the egg. So that's one big difference why we have so many millions of sperms as opposed to such few eggs. Number 1 difference, 4 times more in every cycle. Number 2, whereas the woman is undergoing a cycle once a month, the men are undergoing cycles practically every 24 hours. So it's quadruplicating in contrast with the 1 egg every 24 hours. So at the end of the month, that's how we end up with the millions of sperm that men normally have for procreation.
So question, Father. So in the case where the woman produces 2 eggs, for fraternal twins, where does that second egg come from?
At the very beginning here, instead of having 1 primary oocyte, you had 2 primary oocytes.
Oh, so it's
2 mature at the same time,
okay.
all the way at the beginning. There were 2 primary oocytes that matured simultaneously, 2 follicles. I'll put it into the context in a minute, all right? So it's not that any one of these polar body becomes functional, no, because this is locked in. Remember, nature never forgives. If it's meiosis, it's meiosis, okay? So you have to, if you're gonna get twins, in that case, which would be fraternal twins, right? It was 2 primary oocytes that matured simultaneously, okay? Okay, so let's look at some numbers for our human species. The number of chromosomes, total number of chromosomes, 46 or 23 pairs. But I put it in quotations because strictly speaking, who is the one that has really the 23rd pair, which are identical pairs? The male or the female? The female women because they have XX, we have XY. So strictly speaking, that 23rd pair, the sex chromosomes, are not truly identical for us, okay? In fact, they're not identical at all because that Y chromosome is what makes the embryo become a male, and that X chromosome, which is that X chromosome of the man, is identical to the X chromosome of the women. So that X chromosome has to be disabled. Otherwise, there's going to be ambiguous information to the early embryo. The early embryo is not going to know if both the X and the Y chromosomes from the sperm are functional. Then what's going to happen is the embryo is getting ambiguous signals to become male or female. All right? So the X chromosome in boys, in males, has to be disabled. All right? And has to be, the word escapes me now, if you remember the biologist, but it becomes disabled. It actually becomes all crunched up and up against the nuclear membrane inside the nucleus.
It's called a bar body?
Bar body, exactly, the bar body with double R. And in fact, the same happens for the female because she has two X, but if those two X's express, you're going to get a double female, you're going to get a super female, okay? So one of those two X's also has to be disabled, crunched up into a little ball, into a little body that is called bar body with double R. I think that's the last name of the guy who discovered it.
I think I know some of the super females.
Yeah. We know some super females and some super males, right? Okay, so let's put it even further into the context of the male and female reproductive structures. So here are the testes represented. These are cross sections of diagrams of the cross section. The testicle. Inside are these tubules that are called seminiferous tubules, and that's where the primary spermatocytes reside. Okay, so the inner lining of these tubes, of these little tubules, the inner lining are these primary spermatocytes. Okay, and when they're stimulated by a hormonal process, they start undergoing meiosis, they start undergoing spermatogenesis to end up with the sperm. So here is the meiosis, here's the spermatogenesis of the male, starting with, well, they're calling it spermatogonium here. Yeah, that's the prior step. Spermatocytes, first meiosis, second meiosis, end result is for sperm. And you notice the numbering is interesting. 46 represents the diploid number for the species, in our case, so this is human, 46. And then XY is representing the sex chromosomes, the sex chromosomes. If we follow on the female, here is a cross-section of an ovary, and these are the follicles, ovarian follicles. Each follicle matures, so this is representing like a cycle, on average, a woman's cycle, a monthly cycle of ovulation, of maturation, which, by the way, is a lunar cycle. It is associated with the pull of the moon, the gravity of the moon and water, and on average it's 28 days, beginning with the primary oocyte here, maturing. Here is ovulation or the rupturing of the follicle, and then this remains. This one is known as the corpus luteum. Not to worry, if fertilization happens, this needs to be around to maintain the pregnancy and also to start the development of the breast for future nurturing of the baby.
What's that called?
Corpus luteum. Corpus luteum is what left of the follicle, which also has hormonal functions for maintaining the pregnancy. Okay, so primary oocyte, you see 46 represents the diploid number, and it's always going to be, therefore, an even number. XX representing the sex chromosomes. Then Primary, okay, and what happens, another characteristics in the female is that they start first meiosis and then it's arrested. It's arrested. Second meiosis only happens on the cycle of the maturing of the egg. And so, another contrast in growth and development. Let's say puberty. For the sake of argument, let's put it at 10 years of age for both men and women. We know it varies, and but on average, let's say at 10 years of age. So both the testes and the ovaries are immature for about 10 years after birth, just saying an average time. And at 10, the hormones then finish the maturation. Testicles start producing sperm, semen and sperm. Ovaries start maturing eggs. But what happens is that these eggs were actually formed during the embryonic stage. In contrast with the testicles, what actually took 10 years plus 9 months to mature, in contrast to testicles, the eggs mature halfway in the fetus. In the fetus, okay? As soon as the ovaries start forming, the woman already has a set number of eggs that she's going to have for her life, which is in the millions. So when a baby girl is born, each one of those two little ovaries, talking about normal standard growth, that baby girl inside her ovaries, she has millions of immature eggs which are arrested halfway in the meiotic process. And that's a set number. By the time she hits 10, menarche, her first period, meaning her first ovulation, all right, again, I'm using 10 as a standard number, 10 years of age, she has lost 99% of those eggs. She only has several 100,000 of eggs, all right? So from millions, most of those eggs have degenerated because what? They're old, they're 10 years old. And so they have become too old, over-ripened, degenerate, and what has happened to them? They have been reabsorbed. Nature is also economic, it recycles itself. So the ovaries recycle the old eggs and gets it down from millions to hundreds of thousands only by the time she hits menarche, her first period, okay? Then, but do the math, it's still an overabundance of eggs. Because how many eggs does a woman need really to have a child every, one per fertilization, all right? And let's say that again, I'm just using standard numbers, on average, fertility for a woman, let's say from 10, her first period, which is called menarche, to menopause, which is her last period, let's say she hits menopause at 40. Again, average numbers, they can vary a lot. So 30 years, right? 30 years. If she had a child every month, which is unrealistic and it's impossible, okay? But if she had a child every month, how many eggs would she need? She would need 30 times 12, right? So 3 times 12 is 36, and another 0, 360. So for a woman to have a child every month, or to be fertile, basically, not have a child, but to be fertile, because whether she gets fertilized or not is a different story. But let's say to be fertile, every month of her window of fertility from 10 years of age, which is super young, to 40, which is not that old, all right? But for 30 years, on average, to be fertile every month, she only needs 360 eggs. And she's actually landing at 10 years of age with hundreds of thousands, maybe even tens of thousands. Even 1,000 would be too many, would be 3 times too many, okay? So the vast majority of the eggs in the woman of ova, 99.99% of them die and are reabsorbed by the ovarian tissue, okay? Now, you say, oh, that's a tragedy. No, that's actually a good thing. Because on average, which of those ova, which of those eggs are the ones that are going to be reabsorbed, are going to degenerate and be reabsorbed? On average, the ones that are most damaged. You see, most damaged. Again, elaborate biochemical processes that happen here and detect the eggs that are more damaged. And so it's a quality control. It's an internal quality control that is happening on average on every woman, and not just for the human species. It happens on the mammals. It happens on most species that reproduce sexually. It's a similar process. Okay? So big contrast. And so you can say that in females, the control is done by quality, so that at the end of the whole story, there is one mature egg that needs to be fertilized, and that window of fertilization is 24 hours. 24 hours. For men, for males in general, we go in the opposite direction. We go in the opposite direction. The quality control is in the numbers. And I'm talking about the huge numbers. The numbers, not just in the millions, but in the hundreds of millions. For example, for a man to have what is known as a competent ejaculate, in other words, an ejaculate that is capable of fertilizing, on average, how many sperm should be in the semen, in the ejaculate of a man to be considered functionally potent, in other words, capable of fertilizing, on average? About 100 to 150 million sperm. 100 to 150 million sperm for a man's ejaculate to be considered competent, potent, all right? So you can see that the selective pressure, it only takes one of those 100 million to fertilize the egg, because it's only one egg. And therefore, the selective pressure against sperm is about 100 million to 1, which is very intense, super intense selective pressure, okay? And so on average, which is the sperm that is going to fertilize that egg? The most competent, strongest, most vigorous, best sperm. And so we do quality selection also, but we do it by the numbers. Okay? And why? Again, the reason, form follows function. Because who travels? The egg or the sperm? The sperm travels. And so, because the egg doesn't have to travel, the egg can allow itself the luxury of being large, relatively large, much larger than the sperm, and staying in place, and just one at a time, once a month, thank you. But the sperm, because it has to travel, then it's a marathon race of 100 to 150 million who are competing with each other down to the nanosecond, okay? And that's how the quality control is done in different ways for the purpose, but they're complementary ways, the complementaries. Forward. Okay, I've been going for about an hour and a half, an hour, a little over an hour. So maybe we take 2 breaks. We'll take 1 break now. Then I'm going to go into embryogenesis, actually from fertilization forward. Embryogenesis, the early embryonic development, and then maybe another little break, and we'll look at the ontological status of the human embryo. So this is a good moment to pause just at the doorstep of fertilization, the fertilization process, again at the cellular level. So this kind of summarizes what we've been talking about functionally to end up with haploid sperm or egg, and all the background that it took to get there. Oh, so the number here, bottom line, 23 represents the haploid number, eggs. But you notice that half of these sperm, sorry, important detail that I forgot to mention. Half of the sperm, 50% of those 100 million, 50% are X sperm. Ay, sorry. 50% are X sperm and 50% are Y sperm, correct? Why? Because it's a haploid number, meaning that the 23rd pair, XY, got split. And the X's went to one set of sperm and the Y's went to the other set of sperm. Originally to 2 daughter cells and to 2 daughter cells. But then that was iterated several hundreds or thousands of times to end up with millions of sperm. 50%, on average, 50% will have X chromosome, meaning the maternal X chromosome. Okay. And 50% will have the Y chromosome, meaning the chromosome from the father himself. okay? And so the maternal chromosome is always passed on 50%. And that's another interesting thing that if it is, let's say that it is this, first, let's say the closest one. If it is this sperm that fertilizes this egg, is the result a boy or a girl? It's a boy because there's a Y chromosome, okay? But if it is this sperm that fertilizes this egg, it is a girl. And what is interesting is that this Y chromosome has to come from this father, the owner of this testicle. But this X chromosome, who's the owner of that X chromosome? The mother of the owner of this testicle, the mother of that man. Because a man cannot produce X chromosomes. That came from the mother of the fertilizing man. Interesting, okay? And so that's why we say that X chromosomes are maternally inherited. But Y chromosomes are paternally inherited. And therefore, that's what we can talk about mitochondrial female and Y chromosome atom. Actually, it's a little twist on that. It's the mitochondrial DNA that they're looking at. But anyway, this X chromosome comes from the mother of the man who is fertilizing, okay? All right, good moment to break. Yes, so I'm gonna pause this and pick up maybe about 10 minutes, 15 minutes, okay? Pause recording. Okay, folks, welcome back. Let's continue here. Another bio. We're going to look now at the actual fertilization process. Now that we know a little bit more how the gametes are generated, the egg and sperm, that whole process of meiosis, which again involves chromatic reduction from diploid to haploid, and also recombination, some variety. That's the first level of variety. The other level of variety, for example, comes on which sperm actually fertilizes the egg. So there is variety between male and female, okay? And the other level of variety, now looking at the new individual for the zygote, is that only 50% of his genetic material comes from his father, and the other 50% comes from the mother. Okay, so that's a big introduction of variety there on the new individual, on the zygote. So the fertilization process, the resulting cell, in other words, a fertilized egg, is called a zygote. This is universal, not only for the human, but for all mammals and for all species that reproduce sexually. In fact, not only animals, but plants also. So this is a universal term, making a point about this, because zygote is a universal term. Let's say plant reproduction. And anyone, the first diagram that appears here. If I can find it, there it is, the zygote, okay? Oh, I'm not lying to you guys, I'm not trying to be ideological about it. Just pointing out here is a flower reproduction. So even in plants, the first stage of the individual is called zygote. And from the zygote, guess what develops? The embryo develops. Okay, so these are standard terms that occur in any honest biology textbook throughout the world. Zygote comes from the Greek zygon, and zygon means yoked or linked. And there are at least 3 levels of linkage that will occur, and I'll show you in a minute, or coming together or yoking. It's like the links on a chain, all right? Again, because of egg and sperm, 50% of the zygote DNA comes from the mother and the other 50% from the father of whatever species that is. So let's look at the egg and the sperm. This is a diagram of egg and sperm. Of course, they're not to scale. The scale is rather like this. As far as the structures, the main structures, of course, the nucleus, which is in that arrest of the second meiotic stage of arrest. Excuse me, but the egg is not by itself. The egg comes accompanied with two layers of protection. One layer, which is described here as a jelly coat, is zona pellucida. The first layer outside of the cell membrane, okay, outside of the cell membrane, there is a layer of jelly, mostly proteins and carbohydrates, and it's called the zona pellucida, Latin. And then there is a second layer on top of that, around it, that is called the corona radiata. And these are cells, they are follicular cells from the follicle, all right, from the, remember the ovarian follicle, okay? So the egg comes accompanied. The egg has a cord with it of cells. Here is the egg itself with the zona pellucida and the corona radiata. Important because the sperm, in order to fertilize, has to go through all those layers to empty, in order to empty its content into the actual cytoplasm of the egg.
So, Father, where does the egg open? Oh.
Okay,
Yeah.
I'll tell you in a minute. Because the egg is in fact sealed and closed by these two layers. On the inside of the membrane, shown in these little blue dots, are also these little granules or these little corpuscles, all right? And they're called cortical granules. They are going to cause a chemical reaction on the membrane of the egg, which is known as the cortical reaction, which has to do with calcium. And it essentially seals the egg once fertilization has occurred, so that a second sperm may not fertilize, okay? We'll get to that. I'm kind of anticipating. I'm showing you here the structures and their function. Of course, the cytoplasm is the jelly-like, mostly water, superhydrated area of the egg. Mm-hmm. And now for the sperm has a very different structure, very different shape, because it has a different function. Whereas the egg stays put, the sperm has to travel, all right? So the sperm needs a locomotion machine, which is going to be this mid-region and the tail, the flagellum, which will wiggle and propulse the sperm forward. So it's going to have a dynamic structure, a hydrodynamic shape, beginning with the nucleus, holding the DNA, all right, haploid nucleus. So being haploid is called a pronucleus. Then the tip of the sperm has a little bag here, a little sac that is called the acrosome. That little sac is full of enzymes that are digestive enzymes, because those digestive enzymes are in charge of burrowing through these layers to eventually end up inside the egg itself, okay? And to put it to scale, because these are diagrams and therefore they're not to scale, but this one is to scale. This is an actual macro photograph, okay? So this is a micrograph, a microscopic photograph. Here it has been colorized a little bit to give it some contrast. Otherwise it will be just totally transparent. But this is the egg with the zona pellucida and the corona radiata. And here are sperm arriving. By scale, a human egg is about 1/10 of a millimeter. 1/10 of a millimeter. A millimeter is more or less the thickness of a nail, like a thick, maybe the thumb nail. All right. Anyway, each one of the small ticks here is a millimeter. So just to get a size, pass it around for a moment.
1, 2.
And so it's 1/10. Can't really see it with a naked eye. Could almost see it, but I don't think it's we are able to see that. A cow egg, for example, I think is about 10 times larger. So about 1 millimeter, and a cow egg can actually be seen, a cow ovum can see little tiny with the naked eye. At any rate, the sperm actually is about half of the size, but of course, the vast majority of the sperm length is the tail. And so when you take the tail away, you only have 1/10 of the size again of the whole sperm. So the sperm head actually is very, very tiny compared to the egg. And this would be to scale because that's the photograph. All right, and that's the little bars that I've been showing here. Okay, moving forward. Fertilization is a stepwise process, okay? Beginning with contact. Here is the sperm. The sperm actually burrows through the corona rayata until it hits the zona pellucida. And then the acrosome releases its enzymes and starts digesting through the zona pellucida until the sperm, continuing to wiggle, wedges its way in through the cell membrane of the egg and eventually empties out, the sperm empties out its nucleus with a little bit of the cytoplasm that is there, very little cytoplasm on the sperm, right? Because it's mostly a nuclear package. But that whole content there of the head of the sperm is emptied literally into the egg. This is a standard mammalian fertilization. Humans were no different at the organic biological level. I did mention that the nucleus, of course, of the sperm and the nucleus of the egg are haploid, and therefore they're called pronucleus. Pronucleus. Okay, haploid. And then the two nuclei eventually meet and fuse, the two pronuclei, to form the diploid nucleus. And this is where diploidy is restored. I'm sorry about that. This is where diploidy is restored at the nuclear level, at the DNA level. Okay? So what do we call the nucleus of the sperm? A pronucleus. Pronucleus because it's haploid. All right? And what do we call the nucleus of the egg? A pronucleus also, same term, because it is also haploid. Meaning, what is the N number for humans? What is the N number of the female pronucleus? N is 23. And what is the N number for the sperm? 23. And therefore, when 23 meet 23 and come together, they form 23 pairs or 46, and diploidy is restored. Okay, so by the numbers, you see how it works? Fertilization restores what meiosis split apart. John, question?
So do the nuclear membranes kind of just fuse and come to one?
Correct. So what do we have? That's why that's the zygon. That's the reference to the zygote. The linking or yoking together is really at 3 levels. At the cellular level, between egg and sperm, because the sperm literally empties itself out into the egg. So there's a fusion of cells of sperm and egg. That's the first level of yoking or linking. Necessary but not sufficient. Also, there has to be a nuclear linking or yoking together, the male pronucleus and the female pronucleus, to restore diploidy, to get a diploid nucleus, okay, the fusing together. Again, necessary but not sufficient. The third level of linking or yoking is going to be at what level? At the chromosomal level, at the DNA level. Especially when this zygote grows and develops and matures and is going to undergo meiosis, those tetrads need to link up, those sister chromatids need to link up into the tetrad. So the third level of linking or yoking is going to come at the reproductive level decades after the zygote was formed. okay, but necessary for maintaining the species, the formation of the tetrad. When this individual will become either a man or a woman and will himself or herself produce fertile offspring, fertile sperm or fertile egg. And so you see the three levels of linking or yoking at the cellular level between what and what? Between sperm and egg. At the nuclear level, between the male pronucleus and the female pronucleus. And at the chromosomal level or DNA level, by chromosome 1 of the man pairing up with chromosome 1 of the woman, chromosome 2 of the man with chromosome 2 of the woman, okay? Chromosome 3 of the husband with chromosome 3 of the wife, chromosome 4 with 4, maternal, paternal, all right? So 3 levels of linking, at least organically. There's actually a 4th level of linking or yoking, which is not at the cellular level. Instead of going in, we can go out back, because it's not just the egg sitting here on top of the desk and the sperm sitting on top of the desk, right? This egg and this sperm come from individual humans, an individual man or woman who is an organic whole, who need to have the intercourse necessary for fertilization to happen. So there's a 4th level of linking or yoking actually in the sexual intimacy, in the organic intercourse that occurs between the husband and wife, so that fertilization will occur naturally where it occurs and how it occurs. So really at least 4 levels. You want to take it further, there's a 5th level. If we're looking at holistic view of the human person, it's not just a man and a woman reproducing and then I move on to the next woman and she moves on to the next man. No. We're talking about, if we're going to look at it in the proper way, in the holistic way, in the proper full way, ethically speaking, of a marriage, of a union of life, of a project, of a together forming a project of life. So really, ideally, there would be also a linking of souls, if you will, a linking of 2 hearts coming together to form a life project together, of what could become a family, what could become a marriage for life. So that's the 5th level at the spiritual level of linking. Okay, and that would maintain the physical and the metaphysical integrated into a holistic vision of the human person. This, when finally the diploid nucleus is established within the fertilized egg, right? That's called syngamy. So the whole process of the egg and the sperm fusing and the 2 pronuclei fusing, that whole process is called syngamy. It takes in the human about 24 hours. So the egg is mature for about 24 hours waiting for fertilization, and then that whole syngamy, the whole process as a whole is called syngamy, takes another 24 hours. At that point, that zygote is ready to split into 2 and start mitosis for the rest of its life. Okay? Here is a photograph, a microscopic photograph of a human egg being fertilized by human sperm. And you notice that the sperm actually has to borrow, maybe it's this one, I don't know, but it has to borrow through first the corona radiata and then dissolve the zona pellucida to actually empty out its content. The other thing that I did not mention yet, I'll be with you in a minute, John, is this cortical reaction. This is the counterpart of the female, all right, of the egg. So the sperm is trying to penetrate as deep as it can go, all the way literally into the cytoplasm of the female. The female, for her part, what do you notice here? At least dozens of sperm, and wait another nanosecond, you're going to have another dozen of sperm arriving, because that ejaculate, these sperm came from an ejaculate of, on average, at least 100 million, all right, competitors. And so the egg has to seal itself. And it seals itself literally like a shell, very similar, microscopically, to the shell of a chicken egg, which is calcium, all right? Calcium is involved in this reaction, in that cortical reaction. So these little corpuscles, these little granules, make this reaction beginning and it's kind of a wave function here when the egg actually here at this point when the sperm actually empties its pronucleus in through the membrane, through the cell membrane of the ovum, that starts like a wave action around the egg that seals it in a nanosecond. So that the second sperm that is coming just 1 nanosecond later to do the same thing cannot get in. It's sealed out. Sealed out. So that second sperm can pour out all the enzymes that it wants, the digested enzymes, not going to do a thing.
So
that egg is sealed.
The egg is always willing to receive, but only receive one.
Yes,
one is
once, exactly. Only the first one on average.
That's amazing, man.
It is. It is amazing. It is amazing, and it's very mechanical. It's amazing. Actually, now, so there's a lot of research that's been done. You have to be careful because there's a bias. I mean, if this is a human egg that's being fertilized by a human sperm, then the result is a human zygote, which could be a human being, right? So how are we going to experiment on this? First of all, how are we going to photograph this? Yeah. This, sadly, is coming from in vitro fertilization, which we'll talk about next class. Okay. But at any rate, it's a lot of research because there's a lot of money in vitro. It's a multi-billion-dollar industry, so on and so forth. But the point is that now they're realizing that perhaps the first sperms that actually arrive are contributing with their with their acrosomes. They're actually making holes through the zona pellucida. But it may not be the actual first sperm that makes it all the way through. It may be the second or third sperm that is using that hole to finish the job. Because maybe one acrosome is not sufficient to go through the whole zona pellucida. Yeah. Yeah.
So it might not be the first one that makes it.
It actually may not be. It's a sneaker. We call them the sneaker one. It happens sometimes in fish that the big fish doesn't fertilize the big male. It's the little male that is swimming around. And when the big male is distracted, the little male comes in and fertilizes. It's called a sneaker. It's nature is very interesting, but it's really yeah.
You know what? I think it made it
little tricks.
I was going to teach this morning, and they don't want to.
You see?
No,
maybe they'll watch the video.
Yeah, I totally already.
Yep. Okay, so just to complete the story, you see this little thing hanging out here, okay, stuck there, actually it's kind of wedged in the zona pellucida. Guess what this little guy is? This guy here? That's the bar body. That's the degenerate egg that didn't get to mature because this guy stole all the cytoplasm, and that's the little bar body stuck up in there. The polar body. Yeah. Polar body, I'm sorry, not the bar body. The bar body is in the chromosomes. Yeah, it's the X chromosome. Polar body, thank you. Yep, okay. So interesting, huh? Whole process takes about 24 hours. Let's put it in place, in situ, in place where it happens. Okay, so we're familiar with the female reproductive tract. Here is the uterus and the inner lining of the uterus that is called the endometrium. It's mostly a muscle, it's a smooth muscle. And then the tubes that project out laterally on both sides, and that is the fallopian tube, okay? And then here is the ovary that is connected to the side here of the pelvic floor, the pelvic region by ligaments. But the ovary is a gland that is not really directly attached to the tube, okay? There's a little bit of space here. This area is called infundibulum. Don't have to worry about too much about the detail, but you can think of it a little bit as a glove, all right? It's a glove that is hovering around the ovary. And when ovulation is not occurring, that glove is a little further away. There's a little bit of space there. When ovulation occurs, once a month, on average, every 28 days, on average, with big variation, that fallopian tube, that bend, comes a little closer and brings the infundibulum, brings the glove closer to the ovary. That movement, that physical movement, even if it's just a few millimeters, is real, and that's what causes some of the pain of ovulation, is the movement of those tubes, okay? On average, one on each side every month, alternating on average with a lot of variation. Anyway, here's ovulation, here's the ruptured follicle, and here is the ovum, mature. It stays in this region called the ampulla, which is also known as the horn of the tube or the bend, right? That's the ampulla. It's a little exaggerated. The diagram is not exactly to scale because the ovum would be much smaller than this, right? But here is, and you can see the ovum with the zona pellucida, the corona radiata around it, all right? This region again called the ampulla. This is day one. So for about 24 hours, an average of 24 hours, the egg will sit there waiting to be fertilized, okay? As it sits there, it kind of moves a little bit, but it stays in this general region. It just takes it a little longer to go through the bend. The sperm, if there is fertilization, you see the sperm, it's only showing 4 here of the 100 million. Whoever gets to fertilize first, this assumes that fertilization happened. And now the egg is sealed, 24 more hours for syngamy. And now we have first cleavage. First cleavage is going to be mitosis. Regular cell mitosis, that zygote is going to split into 2, and each one of those 2 daughter cells is going to split into 2 more, forming 4, and each one of those 4 is going to split into 2 more, forming 8, and you see it's a geometric progression, 1, 2, 4, 8, 16, 32, is a doubling of cells. So the number of cells grow very fast, on average every 24 hours, okay? And so that's the process that occurs as that little embryo is traveling through the tube and into the ovary region, okay? It goes through 2 more stages. So the first stage would be the zygote, the fertilized egg. And then the second stage generally is called the morula. And the third stage is called the blastocyst, which is actually what implants, an average 5 to 7 days later. So implantation in the uterus happens in humans, an average of 5 to 7 days after fertilization. It's certainly not part of the fertilization process, okay? A week has occurred between fertilization and implantation. It's dishonest to say that fertilization is not complete until implantation happens. That is dishonest. That is corrupt. And you find some textbooks now trying to redefine biology with ideology and saying, oh, well, fertilization is not complete until implantation happens. That is bad, bad, horrible biology. It's not biology, it's ideology. They're trying to justify manipulating and experimenting on the human embryo because fertilization has not ended.
But it actually occurs in the ampulla?
Yes, normal fertilization occurs in the ampulla.
They have a long way to spend.
Yes, they do, and that's exactly when you look, good point. So if we were to look at the diagram a little further down, let's just look at it for the sake of biological argument here. Female reproductive system. Below the uterus, of course, is the cervix, the circular muscle, and below the circular muscle is the vagina, which is what receives the penis, okay? So these are all muscles. They're smooth muscles. They're not connected to bone, and they're involuntary because they're smooth. They're involuntary muscles, but they'll have their proper function. So this is just a diagram of the female reproductive system. Ovaries attached by some ligaments here. Infundibulum, fimbria are the actual little finger-like structures. The horn or the ampulla, and then fallopian tube, which is also somewhat coil like that. You notice this uterus looks like kind of prepared for implantation because it's thickened like this, all right? And here is that cervix, which is a circular muscle. We have many circular muscles in our body. They're called sphincters or circular muscle. For example, the mouth is a circular muscle, down by the throat, at the top of the pylorus, the muscles at the top and the bottom of the stomach. The anus is another circular muscle, okay? And women have more circular muscles. One is the cervix, which is really the opening to the inside of the body, because frankly, the vagina is exposed to the outside of the body, and that's why women are prone to infections in the vagina, because the vagina is fairly open to the outside of the body, all right? Hygiene, very important. But this muscle is what receives the penis, but the penis doesn't go beyond the cervix. Therefore, semen has to come in through the cervix into the uterus, and that sperm has to travel all the way to here for fertilization. Now, if we compare to scale the size of a sperm, all right, 1/100th or 5/100th of a millimeter, to the tracking that it has to do all this distance, even though in the human body this distance seems small, to scale, it would be equivalent to hundreds and hundreds of kilometers, if you will, to the human size scale. So the sperm has to travel a long distance, okay? In fact, the sperm by itself, right, will only have an average lifespan, again, of about 24 hours. It has very little cytoplasm and very little to live on, just sitting there by itself. But sperm in semen, part of semen is that it has nutrition for the sperm. Plus, in the female tract, also the mucus, the cervical mucus, also provides nutrition for the sperm, so that in the female tract, human sperm can last up to about 5 days. So the actual fertilization could occur 5 days after the intercourse. Let's say they had intercourse before she ovulated, and she ovulated 4 or 5 days later, possibly she could still get pregnant, because that sperm inside the female tract can last up to 5 days. And it takes maybe a few minutes. It may take 5 to 10 minutes to get up there. So fertilization could be as soon as about 5 to 10 minutes after ejaculation, or it could be 5 days later. Those are the two kind of extremes. Okay?
Because maybe if there's an egg that isn't there, but then comes like a day later.
Yes,
there.
that's right. So preparing leading up to fertilization is also important for that window, depending on if the couple is seeking to get pregnant or if the couple is seeking to avoid pregnancy. The events leading up to the fertilization is also important. Therefore, she has to leave herself that margin. Okay? Yeah. So you have to, one thing is to talk about cells, ideally, but cells don't occur ideally. They occur in situ, in place, in a functional setting. And we'll have to think of the functional setting for these cells to be functionally alive. Okay, back here. So you understand this process takes on average 5 to 7 days from fertilization to implantation. What else? Well, about 3 stages, exactly 3 stages to implantation of the zygote, of the early embryo. Excuse me, let's look at those stages. First, the zygote here. These are actual micrographs. These are microscopic photographs of a zygote with the 2 pronuclei are beginning to fuse. You can see here, but you can still see 2 distinct pronuclei. Okay, so this zygote has not completed syngamy yet. It's in the process of syngamy, okay? Because obviously the sperm has already emptied its nucleus, but the 2 pronuclei have not come together fully yet. Once they do, then this cell within 24 hours will move to split. Okay, in fact, it's the sequence here. There's the zygote. And the timing, within 24 hours, that cell has undergone mitosis. And so 1 mother cell, 2 daughter cells. Each of these, as they grow, they split again into 2 more, forming 4, 8, 16, etc., until they form a little mass of cells. These are called morula in general, because now it's the zygote is Greek, but we give equal opportunity to Latin. When these were observed for the first time under the microscope by Europeans that had Latin as a native language, in fact, when I was in FIU in the 70s, studying biology, not even thinking about the priesthood, I was studying marine biology, In the library, I actually found a biology textbook that was written in Latin, like from the 50s and the 60s. Latin was the standard language for science up until just about the first half of the last century. Okay. So morula comes from mora, M-O-R-A, mora, and mora just means berry. Okay, like a raspberry in English, you know, the raspberry with little balls. And so when the scientists were looking at this under the microscope for the first time, the late 1800s, early 1900s, say, oh, mora, but they're very tiny. So they go morula, little mora, tiny berries, morula. It's tiny berry, literally. Tiny berries in there, okay, yeah. So this is a morula stage, and each individual cell is called a blastomere, okay? It's important, it's significant because the blastomeres are identical, they're clones, they're equivalent, and they're all totipotent. In other words, if I dismember this early embryo, if I dismember this morula and put out these blastomeres into individual petri dishes, they, on average, can start undergoing mitosis again and producing new individuals, all right? So they're totipotent, and they, in fact, if they are taken out of their protective coat, then they, in fact, revert back to being zygotes. And this was experiments done on frog eggs by a famous German Hans Spemann way back last century. But the blastomeres essentially have totipotency. They can revert back to being a zygote if they are teased out of they're disaggregated from the cluster. So within 24 hours, we have the first cell division, 48 hours second and third and fourth, and in that geometric progression, you notice that the cells grow very fast, but they maintain the same size in general up to the blastocyst stage. And that's what's also significant about this time sequence here, is not only is there mitosis, normal mitosis going on average every 24 hours, but also you notice that the individual cells, the daughter cells that result, are about, on average, about half the size. So that the overall size of the morula is still about the same size as the original zygote. In other words, what's happening is, at the nuclear level, of course, the nuclei are duplicating or replicating, but the zygote, the cytoplasm is being split in half, okay? So the cytoplasm is being shared with the new cells, and the morula are about the same size still as the original zygote, all right? That is called compaction, because at the level of individual cells, the individual cells are becoming smaller and more compact, and it's a process called compaction. Of course, this is cellular compaction, which is different from the other compaction when I talked about nuclear compaction, which is the supercoiling of the DNA, that 10,000-fold thing that we talked about. That's nuclear compaction or DNA compaction. This is cellular compaction. But it's still a compacting anyway. It's a packaging in. Moving to the third stage, the blastocyst. Okay. At the blastocyst level, what happens? You already have several dozen cells, maybe up to 100, probably not 100 yet, but several dozen cells. And now, two things begin to happen. That little ball of cells, which is the morula, begins to hollow out. In other words, begins to develop a cavity inside, a hollow cavity. Now that hollow cavity is not full of air, of course, because there's no air in there. It has fluid, okay? Mostly water and then other substances in the fluid, but it's a hollow cavity. And a cavity in, forget if it's Greek or Latin, I think it's Greek, it's called a coelom. Coelom is a cavity or a hole. And because it's from the blastocyst, it's a blastocele. So this is the cavity. Also, the cell layer differentiates into two different types of cells. Two different types of cells, okay? The outer layer is called the trophoblast. In other words, on the periphery, let's say, of the ball. That's called the trophoblast. And then there's a little mass of cells. There's an inner mass of cells that develops, like you see here, okay? And an inner mass of cells is fairly distinct from the rest of the cells of the ball. And that inner mass of cells, guess what? It's called the inner cell mass, or ICM, okay? Inner cell mass, or ICM. Sometimes they call it the embryoblast. And why is it called the embryoblast? Because the embryo proper will develop from the ICM. The embryo proper will actually develop from the embryoblast, from the inner cell mass, from the ICM. The embryo will grow from here. And what happens to the rest of the cells, to the trophoblast? The rest of the cells, the trophoblast, becomes the placenta. So you get the beginning of the embryo and you get the beginning of the placenta at the blastocyst stage, okay? And it is the blastocyst who implants into the uterine wall. So going back to this diagram, see at this point, there's a blastocyst, and it is the blastocyst who implants. Here you can see briefly that would be the ICM there, which will become the embryo, and the outer layer like this is the trophoblast, which will become eventually the placenta. Let's go forward a little bit. Okay, so here's a little bit more details, a diagram of implantation. You can see the blastocyst here and the ICM. The blastocyst continues to invade, literally invade and embed itself, has to embed into the uterus of the woman, of the mom. Now, think a little bit biochemically here for a moment, what is happening. The DNA of these cells, this is the uterine lining, right? This is the endometrium that has become thickened, spongy to receive an implantation because of hormonal signals that it has received. The cells, so this uterus is an organ, right? And the organs are made up of tissues. The tissue is smooth muscle, like I explained earlier, uterine smooth muscle, right? That tissue is made up of cells. And those cells are uterine cells. Those cells are uterine cells. They have DNA. Are they haploid or diploid? The cells that make up the uterus. Yeah, because the uterus is diploid, because the uterus belongs to the mother and the mother is diploid. So every cell in our body is diploid except for the gametes. The uterus is not a gamete. The egg is a gamete, okay? So these are diploid cells. Okay, so just to focus on the DNA. The DNA of this uterus, right, of the cells that make up this uterus, come from whom? The DNA? From the mom. So is that DNA 50% of the mother, or 100% of the mother, or 10%, or what? 100% of the mother.
Yeah.
The uterus, you know, her uterus is her uterus, and our testes are our testes. 100% of our testes belong to us, and the DNA in those testes is our DNA. Okay, this embryo, right, was fertilized by an egg that this one produced. But that egg, the nucleus of the egg is 50% of the embryo, right? The other DNA, the other 50% of the DNA of this embryo comes from the man, who is organically is a foreign body. The DNA of that man doesn't belong in this uterus, it's a foreign body, okay? And so here's a conflict. The embryo needs to embed itself into that uterus in order to survive, because it needs a placenta to survive. But the uterus is ambiguous. The uterus only recognizes 50% of that DNA, which is the maternal DNA. The other DNA, paternal, is a foreign body. So the uterus is kind of ambiguous. Should I allow this or should I reject this thing that is trying to embed itself into me? okay? The embryo has already thought about that biochemically, so the embryo is producing enzymes that are blocking the immune system of the mom, so that the immune system of the mom does not reject that embryo as a foreign body, okay? So the embryo is bringing its own defense mechanism, biochemical defense mechanism, to tell the uterus, it's all right, I know I'm 50% foreign, but I'm 50% compatible. So look at my good 50%, look at my compatible 50%, and allow me to embed myself in there, and let's do our thing, which is to connect the capillaries and to form a placenta. Otherwise, you're not going to have a next generation, mom. Okay? So this is all done at the biochemical level. Again, very elegant and very true. Otherwise, we wouldn't be around today. Mm-hmm. So the embryo has to convince the uterus to allow itself to be implanted. And it does. And as it does, it also allows and encourages capillarities, vascularization to happen. So the uterus, in preparation, the endometrium, in addition to becoming spongy, also becomes vascularized. In other words, new capillaries are forming there to assist in the formation of the placenta as that trophoblast continues to grow and develop into, it's called syncytial trophoblast, several stages, but eventually it does form the placenta and the umbilical cord that will feed the embryo. And not only feed the embryo, but also the embryo will pump out toxins, waste, into the mom, and the mom then has to deal with the waste of the embryo through her own system, lungs and kidneys and so forth, to get rid of the embryo's waste. Of course, the good thing is that the embryo is smaller compared to the mom's body. So the mom's body can handle that little bit of waste.
So you have the uterus, right?
Yep.
So all areas of the uterus are succumbing to, can accept the embryo?
Yes, the general, yes, general. But generally it's implanted on the sides, not the upper part. Generally it's implanted on the sides.
To the upper part. Mm-hmm. It does a bit lower than the upper part.
Yes. It has to do, then we get into some issues where the placenta establishes itself. The ideal is for the placenta to establish to either side here. You can see if the placenta establishes at the bottom, ceiling, that's going to be a problem. How's the baby going to get out? The placenta's in the way. Okay? Mm-hmm. So there are issues about where the placenta becomes established. Mm-hmm. And again, they're biochemical. It turns out that this whole inner lining of the uterus has little hairs that move the embryo together with the fluid that is moving the embryo to the proper place. Mm-hmm. Yep. More elegance. Mm-hmm. Yep. So it's all very fine-tuned, and evolution has taken care of the fact that it works because it has gone through so many iterations that the deformed have been selected out. And only the ones who are more or less normal are the ones who have made it through. Okay?
But I've also heard that sometimes it happens inside the fallopian tube.
Yes, that's called an ectopic pregnancy, very dangerous.
Yes.
Because this tube doesn't have, the fallopian tubes or uterine tubes doesn't have enough sponginess to receive an implantation, okay? And so that's the danger. We'll talk about that a little bit more when we get into, I think, NFP or one of the other classes, what to do with a ectopic pregnancy. But it's an issue and it's a concern. Just to ease you a little bit is that a large percentage of ectopic pregnancies become resolved on their own, like they say clinically, medically, they resolve on their own. In other words, typically the embryo dies and the whole tissue is discarded out, all right? Mm-hmm. But when they don't resolve on their own, then it's a danger because that fallopian tube within days starts enlarging and enlarging. If the placenta goes through the fallopian tube, then you have a massive hemorrhage. All this area is highly vascularized, a lot of capillaries, and if those capillaries are breached, massive hemorrhage for the mother, she could lose her life. And so ectopic pregnancies are a concern. We'll deal with them in a few classes when we talk about NFP. Mm-hmm. John.
Yeah, the 5 joining that you mentioned ideologically.
Oh, okay.
And then there was, sorry to go back for a second.
Mm-hmm.
the actual man and the woman,
The linking?
and then the actual act, right?
Yeah, so the actual intercourse, right? There's
and
a
then the egg
linking.
and the sperm and then the DNA.
Right.
And then is it safe to say that some of those 100 to 150 million sperm would go to the wrong fallopian tube?
Exactly.
DNA? Yeah.
How many? On average, 50%. And of course, we know that men don't ask for directions, so those are lost. Exactly. So right there, you have the chance. You're down. Now you're down to 70,000, 70 million. Yeah, exactly right. Okay, gastrulation. So the fourth stage, once the embryo embeds itself into the uterus and the pregnancy has been established, all right, the little embryo will continue growing. Remember the ICM? And, oh, by the way, I want to mention that at the blastula stage, at the blastocyst, is where we have the first cellular differentiation. In other words, we have two different types of cells for the first time, right? Because the blastomeres were all equal. Here in the blastocyst, we have an inner cell mass, which becomes the embryo, and then we have the trophoblast. So we have two distinct cell layers that have two distinct functions. The ICM will not become a placenta, and the trophoblast will not become an embryo. Okay, so this is the first moment of cellular differentiation. First moment of cellular differentiation. That cellular differentiation will continue to progress into the next stage, which is known as the gastrula or gastrulation. And at gastrulation, a third layer comes onto the scene. This is what happens. We start with the blastocyst here. Here's a trophoblast forming the ball, the hollow cavity, and the ICM, right? As this little embryo has embedded himself or herself, development continues, and two cell layers appear. There is the ICM, actually develops into, differentiates, the ICM differentiates into two cell layers. One inner layer that is called the ectoderm, and then the outer part, the endoderm. So we end up with two cell layers from the ICM as the ICM continues to differentiate. Two cell layers. As the embryo continues to develop, the ectoderm, okay, the upper layer, if you will, the ectoderm, starts pouring in between the two layers. It's pouring in. That process actually is called an invagination, because a vagina is an infold, okay? So that process of pouring in is called an invagination. Too much detail, don't worry about it, but the point is that the ectoderm pours in between the ectoderm proper and the endoderm, forming a middle layer, and that middle layer is called a mesoderm. And so the end result, just stay with the big picture, is that at gastrulation, we end up with three cell layers. An early embryo that has three cell layers. Has an ectoderm, has a mesoderm, and has an endoderm. The derm is a reference to cell layer, okay? And ecto, meso, and endo is just a reference to their location. These three cell layers are known as the germ layers because all of the organs and tissues are going to develop from these three cell layers in categories. All of the organs and tissues of the body are going to develop from these three cell layers. For example, the skin. Where do you think the skin develops from? Ecto, meso, or endo? Ectoderm. The skin, the ectoderm becomes mostly skin, okay? Which is an organ, it's a system, right? The integumentary system. Whereas the digestive tract and many glands inside the body come from the endoderm.
Glands?
glands and digestive system, the digestive tract come from the endoderm. It's kind of like a tube on the inside of the body. And the mesoderm, you have things like the muscle, skeletal system, muscular system. So the different organs and systems of the body will come from these three germ layers as they continue to differentiate, become more and more specific according to a particular function. So the development of these three cell layers is called gastrulation. And the end result is a gastrula. So we went from zygote, single cell, to morula, several cells all identical to each other, blastomeres, to blastula, or blastocyst, where we have two cell layers, an inner cell mass and a trophoblast. Now, the first stage, the gastrula, where we already have the three germ layers established. And there's a groove in the middle, okay? And the embryo is becoming more and more elongated. So it will have an anterior part, which will become the head, and it will have a posterior part, which will become the rest of the body from the neck down. So there's an orientation, anterior, posterior. And it is symmetrical, left and right side. Looking a little bit more in detail, don't worry about all of these names. The most important thing is the chronology here and the timing. This is gastrulation. This is a cross-section of uterine wall with implantation occurring. So the pink color here is the endometrium, the uterine wall with all the crevices that it has, cisternae. So the sperm, for example, had to travel through all these crevices in and out, in and out, in and out, in and out throughout the whole uterus to get up to the fallopian tube. That's why the distance is magnified tremendously. Okay, so here's an early blastocyst implanting. Here's the beginning of the placenta, what I mentioned, the syncytial trophoblast, not to worry about the terms. Here are the germ layers establishing, transitioning from the blastocyst to the early gastrula, becoming more established, and now fully established. Gastrula is fully implanted, all right, embedded within the endometrium, and we are here at about 12 days. So the second week on average. The first week was from zygote to blastocyst, first week, and from the ampulla to the uterine wall, implantation, and the second week is that process of implantation and gastrulation. So by the end of the second week, we already have that gastrula. And here is a micrograph. This is an electron microscope photograph of a human gastrula. You can see the anterior posterior orientation, the neural groove. It's called neural groove, which, by the way, that what happens with that invagination of ectoderm, as the ectoderm pours in, generates a little tube inside, and that tube is going to become the neural tube. That tube is going to become the spinal cord, the nervous system. So it's interesting that the nervous system also comes from the ectoderm originally. The nervous system comes from the ectoderm, and yet the nervous system is the innermost system of our body. It's inside the cranium, inside the skull, it's inside the spinal column, it's inside the vertebrae. How did it get in there? Well, precisely by this pouring in of cells, by this invagination process, through the groove, through the neural groove, all right, and differentiating into neural tissue. So the skin and the neurons have a common origin in the ectoderm. Talk about specialization. Very different functions, very different locations. One is on the outermost part of the body, the other one is on the innermost part of the body. So a lot happens in the embryonic stage, in the early embryonic stage, 2, 3, 4 weeks. It's amazing how much happens for that little embryo to be formed. All right? So this is a cross, you can see the 3 layers here. The outer layer is the ectoderm, mesoderm in the middle, and the endoderm in here, which will eventually become like I said, digestive tract and other glands and so forth. All right. Another level. Going forward. This is a series called the Carnegie stage. This started last century sometime, and this has been classified. It is kind of the standard staging of development for the human embryo. And it's done by the Carnegie Foundation, which starts with a zygote here. All right. Of course, not to scale. This is much larger. You see that the two pronuclei are still trying to meet there. So syngamy has not completed yet. You can see a little polar body on the side. Then just a little dot represented. Day 1, 2, moving forward. These are stages because of events that happen biochemically and structurally and functionally that have been classified like this. As we go from the zygote through the morula to the blastocyst in plants, and then we get the gastrula. Here you can see gastrulation. Maybe if I make it a little larger. Okay, it's beginning, see how it has that orientation. Anterior, posterior. Look at this structure here, right? Somewhere like this. You can see the head region is on the top part, and it's bilateral. You can barely see the groove, the sulcus. Continues to move forward. We're at now at 3 weeks here. It's an advanced gastrula. Early embryo continues to grow and much more differentiated. The head can be seen. A tail can be seen in the human. There's a human embryo. Wow, look at that tail of a human embryo. Where's the tail? It's a vestigial tail. What's left of the tail is the little tail bones, the coccyx that are fused. We don't see it on the outside, but you fall in the back and you break that little tailbone, you'll know it, you'll feel it, and you'll know that you have it, okay? So that's what's left of the tail is reabsorbed, you see? It's called a vestigial structure and is reminiscent of common ancestry. We go back 5 to 8 million years ago, and we find a common ancestor with chimp and other primates. We go back another 5 to 10 million years ago, we find common ancestry with other monkeys who do have still the functional tail. All right, so mammals, most mammals do have a tail. Think of zebras, elephants, tigers, etc., etc. Most mammals, terrestrial mammals do have a tail, right? And we used to. It got reabsorbed. So remember, ontogeny and phylogeny. You also see the limb buds. They're called limb buds. This is the forward limb buds. This is the rear limb buds, B-U-D-S. A bud will eventually become a leg or an arm, and then the digits will begin to develop. Digits meaning fingers or toes. Okay, so you see it's gradual but definite development, and it's a one-way development. We're not going backwards on this. The cells are becoming more and more specialized. They're growing. The tissue and organs are growing and growing. You can see the beginning of the eye forming. Okay, continues to grow. See the placenta here, truncated. I'm sorry, the umbilical cord. All right. Continue. You can see the head is disproportionate to the rest of the body, almost half the size of the body, because the brain has to grow a lot fast early so that by the time baby is born, the brain has enough development to sustain the rest of the life of the baby and the infant, because the brain is controlling everything that's happening in that organism. Okay, so the Carnegie stages, let me go back for a moment, zoom down, are known. Standard is a standard in biology. There are 23 stages from zygote to 8 weeks, development. By the time we get to 8 weeks, the vast majority of the organs and systems of the body are formed in place, but in miniature, because this little embryo is about 1 inch long. Okay, so by 8 weeks, The vast majority of organs and tissues of the body are formed in place, but the whole little embryo is about 1 inch long. And so the development changes name from embryo to fetus, exactly, which in the Latin, fetus means tiny bean, fetus, all right? So up to 8 weeks, here in this scale, the weeks are shown in this first horizontal line here, row. 7 weeks, 8 weeks, to 9 weeks, changes a name from embryo to fetus. It's a technicality. The embryo doesn't know that. The fetus doesn't know that. The embryo and fetus are just continuing to grow in a continuum of growth and differentiation and actual. So by this point forward, from the 8th week forward, it's mostly growth in size, The brain, the liver, the heart, the legs, everything is growing in size, which is making that fetus bigger and bigger and bigger until finally it can't stay in there any longer, has to come out. And so the first set of diagrams, the first set of caricatures here goes to 12 weeks, which divided into 4 is 3 months, right? So this would be the first trimester. So these are weeks, 7 week, 8 week, you see, 9 week, 10, 11, 12 weeks, or 3 months, first trimester out of a total of 9. So by the time you get to this scale down here, the jumps are in months. So we go from weeks to months here, 4 months, 5 months, 6 months, because otherwise it wouldn't fit. And basically, by and large, what is happening to the fetus is the fetus is growing in size. Is growing in size internally and therefore also externally, all of the organs and systems. But again, by the 8th week, the vast majority of those organs and systems are already in place, but miniature. So we go here from 1 inch at 8 weeks to about 1 foot on average for a baby ready to be born. Mm-hmm. When we consider that size, actually, from the zygote, which was, the zygote is the size of what? The zygote is a fertilized egg, right? So the zygote is about the size of an egg, of a human egg, and that human egg or zygote is 1/10 of a millimeter, 1/10 of the thickness of a nail, to about 1 foot long. I did the math one time, more or less, how much growth occurs within 9 months, less than a year. The growth, just by the numbers, is about 5,000 times bigger. From the zygote to a newborn baby, just by the numbers on the size, it's about 5,000 times more in less than 1 year, right? Now baby is born. And let's say by the time that baby becomes an adult, let's make it a tall adult. Let's say that the adult is 6 feet, right? And let's say that it took maybe 20 years to get there, 15 to 20 years. From 1 foot to 6 feet, took another 15 to 20 years. And then from 20 years forward, 20 years of age, basically we stay the same. And in fact, after a while, we start shrinking, right? When we shrink, we lose calcium on the vertebrae, and the vertebrae start collapsing and getting shorter. And that's how we lose height mostly. It's by loss of bone mostly. But anyway, let's say we stay 6 feet. So it took less than 1 year. It took 9 months to grow 5,000 times. And then it takes the rest of our lives to only grow 6 more times. Totally disproportionate, right? Totally disproportionate time to growth. What I'm trying to emphasize is that embryonic development and fetal development is incredibly fast, very fast compared to the lifespan of a human being, and this is standard. It's about the same gestation period for most mammals. I think whales are maybe a year and a half. It varies a little bit by a few months back and forth. Okay, but it's totally disproportionate. What happens during the embryonic and fetal stages and period is extremely fast, very fast. compared to the rest of the born life of the individual. Okay? Therefore, fast means delicate, precision, anything can go wrong. What goes wrong early will make a big difference later on. A few cells mismatch at the beginning will make a whole tissue dysfunctional later on, okay? And so embryonic development is delicate, and that's why it's inside, literally in the innermost part of the female for mammals, because we also produce fewer offspring. You know, we don't have hundreds of thousands of offspring like frogs and fish and other animals. Because we have fewer offspring, they are more protected, and it is not only internal fertilization, but also internal gestation, namely the pregnancy. So what nature is trying to do, nature is trying to protect this embryonic growth and this fetal growth as much as possible, literally by the body of the female, by the body of the woman, in her innermost part, which is basically where the uterus is located. Okay, so you see again, the functionality, form follows function, right? The structure of the thing, the shape of the thing is according to the function of the thing. Okay, we are now at a little after 12. We're in good time. We can take a break if you need a break, or we can continue. Basically, what we're going to do is go into the implications of all this biology. What does it mean for bioethics or for the ontology, for the status of the human embryo as human specifically? Because up until now, what I've been saying about this organic development is standard procedure for any mammal that, that reproduces sexually, right? Right, so first, questions, comments, anything? Follow along, right?
You said from zygote to blastoplast first week.
Yep.
Blastocyst
Yep.
to implant.
Through implantation to gastrula, second week.
Okay.
Right?
And then to gastrula.
Yeah, to the gastrula.
Secondly, and gastrula to?
Then gastrula to, let's say gastrula to the 8th week. Right, is the embryonic development to all the organ systems in place.
Okay.
And then from the 8th week forward is the fetus, mostly grows in size. So it's multiplication of cells. So really from the zygote forward, it's mitosis. Mitosis and mitosis and mitosis. And this little embryo will not see a meiosis until when?
So it's reproductive age.
Exactly, to reproductive age. Maybe on average, 10 to a dozen years after birth, when his testicles become functional, start producing semen, or her ovaries also become functional and start ovulating, all right? So it's all mitosis. Just cell division, cell growth, development, specialization of the organ tissues, and so forth. You want to take a couple minutes? Yeah, okay. Shorter, about 5 minutes or less, okay? Let me pause for a moment. Pause. Okay, welcome back for this final route here, about 15 minutes. Kind of short, but I think we can do it. We're going to look at the ontological status of the human embryo at the level of the zygote. Okay, so we're going to go back from baby, back 9 months to the origin of it all, when that egg was fertilized and became a zygote. Which leads me again to the words semantics or etymology. Etymology is the origin and the meaning of the word. And semantics is the meaning and how the perception, how words can also be played around with. Okay? So to give you an example of what I mean is words make a difference. And words have meaning, and it's important to use the right word. We hear all the time fertilized egg. Fertilized egg, fertilized egg, fertilized egg, yeah. But we consider it technically an egg. Is an egg diploid or haploid? Haploid.
Yeah.
It's an ovum, Human ovum came through gametogenesis, came through meiosis, came through oogenesis. Therefore, the egg is haploid. A fertilized egg by definition is diploid. And so a fertilized egg is kind of a misnomer. Fertilized egg is really a zygote. Okay? So when you hear fertilized egg, it's just placing, it's kind of ignoring the sperm. In a sense, just placing emphasis on the egg. But the egg is no more. Because once an egg is fertilized, there's no more egg. We have a diploid cell. Even before syngamy, even before the two pronuclei are fused, once that male pronucleus, once the sperm pronucleus is poured into the female, by definition, it has become a diploid cell. So even fertilized egg is already a misnomer. It can give the wrong connotation that we're just talking about an egg, which is not a full human being. An egg is an egg, just like a sperm is a sperm. But a fertilized egg is a zygote. And could it be that that's the beginning of a new individual? Okay, so that's what I want to look at. So is a fertilized egg, in other words, is a zygote an individual human life? Right? I will go back to that little plant. Okay, here is again this plant, which is too big. Sorry. All right, so I just went on the web and pulled out the first diagram that I found on plant reproduction, which is the flower. You know, the flower is the sexual organ of a plant, and it's a complete organ. It has male and female structures. The male structure is known as the stamen. Don't worry about these names, just giving you the background. And that's where pollen is. So the equivalent of sperm in plants is pollen, right? All that yellow pollen that you see flying around, that's the equivalent of sperm. In fact, each pollen grain contains inside 2 sperm cells, 2 sperm nuclei, all right? You see them here, sperm nuclei. So the stamen produces the pollen flies around until it attaches on the stigma, which is part of the pistil. The pistil is the inner part of the plant and is the female structure. So the stamen is the male structure and the pistil, the inner part of the flower, is the female structure, all right? Pollen gets stuck up here and it sends out these little tubes called pollen tubes that go through the pistil, the tissue, until it hits this lower part. And this lower part, guess what? It's called the ovary, because down here in the ovary are the ovules, or eggs, or ova, all right? So this is standard terminology. It happens in plants, it happens in animals, any species that reproduces sexually. All right, so once the tube is formed, then down come the nuclei, the 2 sperm nuclei. In the case of plants, it's 2 nuclei instead of 1, all right, because one nucleus fertilizes polar bodies and the other nucleus fertilizes the actual egg or ovule, and it forms a zygote. What I'm trying to point by this is that you see in a standard biology diagram, any species, plant or animal, that reproduces sexually, the first resulting cell is a zygote. And from the zygote grows the little embryo that you can see there. Okay? Now, let's say that this plant looks like what? Yeah, maybe even more lily, like a tiger lily. You know the tiger lily? Okay? All right. But anyway, it's a plant. It's a beautiful plant. It's a beautiful flower. This little embryo here, as this embryo grows, and here the embryo inside the seed germinates, and the little seedling comes out, right? That seedling grows and continues to grow until it becomes a mature plant. What do you think is going to come out of here? A giraffe, alligator, butterfly? It's going to come a plant. And the plant is not going to be just any old plant. It's not going to be a pine tree. It's going to be a tiger lily. All right, so no one questions that this little seedling is the seedling of a tiger lily. And this little embryo is the embryo of a tiger lily, because that's a species. That maintains species. There's no way that another sperm cell, another sperm nucleus from another species could fertilize this tiger lily egg. Won't happen. There's no compatibility. Okay, so if that is true for other animals that reproduce sexually, why do we become so ignorant and all of a sudden question out of the clear blue, oh, is it truly human? Well, if the sperm is human and the egg is human, that zygote has to be human. Otherwise, it's not going to happen. You know, a chimpanzee egg is not going to fertilize, is not going to be fertilized by a human sperm, and so forth. So, my proposal is that a human zygote is human life. And it's individual because it's one. From that zygote, every time, normally, when we leave it alone, a new individual comes out. So, individual human life. It's just applying some basic reasoning to the biological facts. Of course, we have to know where the zygote came from. And we have to know the conditions of how zygotes are formed from the fusion of gametes and of the same species and so forth. So how about is this individual human life a human being? Okay, so we have to look at the word. We established that it is human, but is it a being? Well, everything's a being. Everything that is a being. This is a bottle being. This is a foam being. All right, this is a paper being. Anything that is a being is a thing. It's an existence. All right. So if we're looking at an embryonic elephant, we can say that that's an embryonic elephant being. All right. Or because the being is just saying that is the essence of human. That is substantially human. To me, the final question is that zygote a human person? That's where most people, in my opinion, that's where they get stuck. Yeah, but is this little cell, this tiny microscopic cell, 1/10 of a millimeter, is that really a human person? Like you and I are human persons? Challenging. Again, all we can use is reasoning, because if we don't use reasoning, then we use emotions and passions. All right? So we know definitely it's not a legal person, because a human embryo can be destroyed legally. Nobody gets arrested for having an abortion or for performing an abortion. They used to, but it stopped in 1973. In fact, yesterday we commemorate, sadly, once again, from 70, what is it, 45 to 8, 3 and 8, 6, 46 years. Estimated to be 60 million abortions in the United States alone. 60 million, that's more than all the wars in the world put together, just in the United States. And so it's significant that the human embryo is a human embryo, human person, because certainly it's not a legal person. It doesn't have protection under the law. Okay. So where does person come from? Greek again. Person comes from prosopon. Prosopon in Greek means the mask. So we have to go back to Greek theater, the mask. Okay, in classical Greek theater, here's an amphitheater somewhere in Greece. This may actually be southern Italy somewhere, where there was part of Magna Graecia, the part of Greece. But this is an amphitheater, and you notice the design is very intense, very specific. It's a semicircle. It slopes up gradually at a specific angle, not very steep, rather low, shallow angle of rise, okay? And this is definitely the stage area with the circular platform in front. So what was represented in the Greek theater? What was represented in the Greek theater was Greek mythology. What was Greek mythology? Greek mythology was polytheistic. In other words, they believe in many gods, several gods. And the gods were not necessarily rational, they were very emotional. And they were having fun with the humans. And they would torture the humans, and they would persecute the humans. Sometimes some male gods fell in love with female humans and had sex with them. And what developed from that union of a mythological Greek god mating with a beautiful woman, what came out of there, the result were the heroes, like Hercules and Ulysses, and heroes looked human, but had superhuman qualities. Because the humanity came from the mom, which was human, but their superhuman qualities came from the dad, which was superhuman, was a god, right? And those are the heroes. And that's Greek mythology, which is very sophisticated, very elaborate, and so forth. So Greek theater, this is what a civilization does when they're not at war with each other. They actually represent their religion in stage, in acting, okay? And Greek theater started by the representation of Greek mythology, which was a common knowledge. Everybody knew the stories about Zeus and Venus and Jupiter and Mercury, right? Because it was their theology, it was their religion. This is a mythological religion. Okay, so typically there was one actor. And the one actor, who was always a man, had a huge voice, had a very strong voice. In fact, I've heard that they would train, to be an actor was, you know, you were on top of the line on society back then if you were one of these actors. And you would train, you would go to the sea coast, to the Aegean Sea, at the Mediterranean, there on the coast somewhere, and you would put a few pebbles in your mouth and say the phrases, the oratory, whatever you were going to say, with the pebbles in your mouth, and you practice against the ocean, against the waves. And that's how they developed their thoracic capacity. That's how they developed their projection. Because there was no microphone. Okay, there was no microphone back then. And so that guy had to project that story out for whatever length of time it took into this theater, this amphitheater. But the theater is designed in a way that if one speaks here loudly enough, everybody hears, all the way up here. Okay, look at the size of that little creature. People sitting up here, these are people sitting on the last rows, on the last pews. Okay, they could hear as long as the guy projected because the sound amplified and carried out. This is Greek architecture, which is based on...
churches today.
Yes, exactly. The amphitheater, to this day, 2,000 years later, the amphitheater is the best way to transmit that sound in an open setting, okay? So the acoustics were put together by the physicists, by the engineers who designed these things, and this was classical Greek theater. Back to the mask. So this one guy, he would typically sit there, and in front of him, he would have a table like this. And on the table, there will be the different masks of the different gods. Now, the audience knew what mask represented Zeus, what mask represented Jupiter, what represented Bacchus, you know. They knew what mask represented different, this is obviously a female mask, all right? That's probably a very upset man there, male god, etc. And in fact, it was the two types, right? So the original Greek theater was actually tragedy. It was tragedy, okay? The angry, the one that's the mouth is facing down. Why? Because it was a fatalistic society. In other words, these humans, they had no way of getting out of the oppression of the gods. The gods had control over them. All right? That was the Greek mythology. We call it that it was fatalistic in the sense that they had no way of breaking out away from the gods. And that's why the heroes were so important, because the heroes, by doing some works, they could gain some favors from the gods. Okay? But anyway, the original Greek theater, I understand, was tragedy. And then what happened was wine was introduced because there was one god, was the god of grapes and the god of wine, which was Bacchus or Dionysus. I think Dionysus in Greek and Bacchus in Latin maybe, right? And so when wine was introduced into the theater, people started getting happy. And they didn't care less if they were in a tragic, in a fatalistic situation, they got happy. And so comedy was introduced. And then the mixture between the two, then you get drama, all right? Which is more, you see how it's getting more and more realistic about life, the drama of life. You know, we have some happy moments, we have some sad moments. Anyway, back to the mask, which in Greek is called prosopon. That is what represents the person. So the mask does two things, conceals and reveals. The mask reveals the protagonist, reveals the god or goddess that is being represented with that line of oratory or phrase. So the mask reveals the god, but at the same time, the mask conceals the actor, right? Because who cares what the actor looks like? What I want to know is what god or goddess is being represented. What line am I hearing from which goddess or from which god? What part of the mythology? I don't want to see the actor. I want to see the god or goddess that is being represented, or maybe the hero. And so the mask simultaneously reveals the personage, reveals the, what is it called? The protagonist, what is it called in a movie? The actor? The person that I'm acting for, or
Character?
the character, thank you, the character. So the mask represents the character, reveals the character, but conceals the actor, okay? And that's what person comes from, prosopon. And so to apply to today, what is the mask in us?
Our ego.
Yeah, okay, but even physiologically, we start with the physical, the mask is the face. The face simultaneously reveals and conceals. So I can reveal some emotions, right, with my face. We have a lot of muscles over which we have control in our face, but also it conceals other thoughts. You know, no one knows what I'm thinking except two people, myself and God, right now. I can be thinking any kind of things, and it's concealed because the face reveals something but conceals also something. So the face simultaneously, the countenance simultaneously reveals and conceals, okay? But if we take that person in the fuller extent, okay, when we look at zygote, okay, where is zygote? Oh, here. All right, this zygote simultaneously reveals and conceals. The zygote looks like a cell, like a single cell, round, circular. It looks like an egg, actually, has the same size. It even has the zona pellucida and corona radiata around it, but is concealing the DNA. The zygote has to be diploid to be a zygote. And that diploidy comes from the fusion of two gametes, of two pronuclei, the male and the female pronuclei, of the same species that have to be compatible with syngamy and so forth. And so the earliest stage that we can go to the individual, the new individual, is truly the zygote. If we go past the zygote, we have an egg that belongs to the mom and we have a sperm that belongs to the dad, okay? So what I'm proposing is that from one perspective, anthropologically, ontologically, philosophically, the first person is the zygote because simultaneously reveals and conceals. Reveals that he or she is a cell that if left alone will continue to divide normally in the process of mitosis, and 9 months later a baby will come out from there. And at the same time is concealing within the nucleus his or her identity as human in the DNA, in the 23 pairs of chromosomes. And so my point with all this is simply that if we understand person and the etymology of the word, from one perspective, the zygote is the earliest that we can attribute ontological personhood. Not legal, because we know that, so what happens, because the zygote or the human embryo actually at any stage is simply not mentioned in the Constitution, in the US Constitution. And that's why it doesn't have legal protection. And Scalia said it for many years. Judge Scalia, one of the Supreme Courts, practicing Catholic who died, said it for many years. Now, you know, the judges of the Supreme Court are experts, supposed to be experts on the US Constitution. He said, it's simply not mentioned. The human embryo, the unborn, is not mentioned in the Constitution. And so it depends. If we interpret the Constitution traditionally or conservatively, we don't add anything to the Constitution, the US Constitution. If we interpret it liberally, then we say, well, because the human embryo is not mentioned, then the human embryo is not a legal person, and we can kill the human embryo. And you tell me a newborn it's illegal to kill a newborn, right? Newborn baby, I have a newborn baby here. We protect newborns to the hilt as soon as they're born. But 1 minute before they're born, can you tell me what difference is there ontologically or biologically? 1 minute before, 1 hour before, 1 day, 1 month, 9 months before. When do you draw the line? Every other line, as I have tried to show you, is a continuum. It's a continuum. And so the earliest possible is the zygote. Okay?
Before in reference
I
to
rest my
what you
case.
said about not that all babies get protection.
Sorry?
You mentioned
Yeah.
something to the effect that all babies, human life get protection.
Right.
Actually, I think the House just passed a bill. But
Yes.
it wasn't passed before.
Yes.
Because remember, if in case of a botched abortion,
Right.
and the baby comes out whole, they leave it on the table.
Yes.
There's no protection. No,
Right.
that cannot happen anymore.
That's right. That was proposed, but it never went through. So
Yeah.
just
Well, there
yesterday.
was one president who actually was a senator that voted 3 times against that one bill.
Yes. Yes. Mhm.
I won't call names, but
Exactly. Well, we know how the parties are divided and all
Yeah.
that, and that's the issue. But as recently as yesterday,
Yeah.
that bill that you're mentioning, which is known the Born Alive,
yeah.
short for this short term, is called Born Alive Bill. All
Yeah.
right. It passed the U.S. House and Congress, which means that if abortion goes wrong and the baby is born alive, then that same team that was trying to kill that baby a minute before has to do everything possible to try to save the life of the baby, because now that baby is outside of the womb and therefore has protection under the law, has all of a sudden become a legal person
So that happened.
just by coming out, by the head
Just-
coming out.
just yesterday.
So it used to have protection.
They never
That's
had it.
right. And there was a conjure about that because many of us thought that that embryo did have that fetus did have protection, but no,
But
it
simply
was
because
never
it
a law.
it was an abortion case.
Yes.
And it was born, it didn't have protection before.
Right.
It was
Correct.
an abortion that went wrong. That's what you're
Yes,
saying.
yes, if it's an abortion.
Because
yes.
the statement is that the baby survived, but having that baby live is against what the mother wanted.
Right.
Because it's the mother subject.
It's against the action of the abortion.
The abortion gets botched.
Mhm.
So the mother didn't want a child.
Right.
So we
So
should.
the child could be
protect that
left
child.
there to die, and it actually happened. Right.
That's terrible.
So now it's gone through the House.
Yeah.
Another thing is the Senate. So we have to
Senate.
pray for the
It's not
Senate
trouble in the Senate.
to have a companion. It's called a companion bill.
Yeah.
Because that's what President Trump announced yesterday in part of his little speech that he said to the pro-life march, that when that bill comes, if the bill comes to his desk, he'll sign it. All right. And so that's where we're at right now. Yes.
Do something good.
Mm-hmm. Right. So, okay, so I've tried to give you first the biological background for when human life begins, and then the ontological reasoning for being consistent. This is what we call an argument of logical consistency. If that individual is a human life, then he or she will be such from fertilization, from conception, okay? One last thing, if I take a couple minutes to finish this, please, is then the language, because I'm saying that words do make a difference. Calling the human embryo an it is a contradiction in terms. And in fact, pro-lifers, even I've been doing it in this lecture, just for the sake of simplicity, I've been calling this gastrula and this morula and this blastocyst, I've been calling them it. It implants, it this, it that. But technically speaking, strictly speaking, we should never call a human embryo an it from the zygote forward. Because what are it? It is a bottle, it is a phone, it is a computer.
Yeah.
So it's are things. It is a ruler, but human beings were not things. We are either him or her, you see? And that's when we begin, when we call the human embryo an it, we are objectifying the embryo. We are dehumanizing the embryo. And it's difficult. To this day, I've been pro-life for decades. I have a difficulty, I get caught sometimes in referring to the human embryo as him or her. But you try referring to the human zygote forward as him or her, and you'll see how it catches people's attention. He implants, for example, or she implants, or she starts embryonic development. But then when we use he or she, we are humanizing the embryo, which is the proper thing to do. That is what I call intellectual integrity, intellectual honesty. And that's where we challenge people. Thank you very much. And God willing, I'll see you next Friday, next Saturday.
Thank you very much. That was
Okay.
good.
I'm going to shut it down now and start the download.